All right. Good morning, good afternoon, and good evening, everybody, wherever you are on this wonderful planet. Welcome to the AWG Workshop 2021. I just wanted to take a moment and go over some housekeeping items before we jump into the opening remarks. So for today's workshop, I will kindly ask that all the registrants, all the participants, please type in their questions in Conference IO, any questions you may have for the speakers. So for those of you that have not used Conference IO before, this is what it looks like. And there will be a plus sign here to the right-hand side. And if you click on that, you will be able to type in a question. Now, the caret on the left-hand side here allows you to upvote any questions that you may like. I will do my best to answer as many questions as possible in the allotted time, but sometimes we do get cut short on Q&A, so I just wanted to let you know at that point, I will prioritize based on the number of upvotes for any particular question. For speakers, I just wanted to send you a friendly reminder that you will receive 1 ding of a bell for your 2-minute warning. If you hear 3 dings of a bell, that means your time is up. For the lightning talk speakers that have 5 minutes to speak, you will get a 1-minute warning with 1 ding of a bell, and you'll also hear 3 dings of a bell when your time is up. So with that, I would like to introduce you to Jonathan Galaska. He is the GeneLab project scientist, and John obtained his PhD at the University of California, Berkeley, and became the NASA GeneLab project scientist in 2017, joining the Space Biosciences Research Branch in 2015. Before this, he was the NASA postdoctoral fellow program at NASA Ames Research Center, studying the genetic and epigenetic response of yeast to microgravity exposure, and a postdoctoral scholar at Oregon State University, where he studied the mechanisms of heterochromatin establishment and the role of heterochromatin in maintaining genome structure. Marco, if you can unmute John so he can give the...
Hi, working now, thanks.
All right. So John, go ahead and take it away.
Great. Well, thank you, Lavorka. And before I start, I just wanted to recognize Lavorka and Marco Bolt and KBR for supporting this workshop and making it all happen. And I'm going to give you a bit of context into this showcase. Next slide, please. The NASA Space Biology Program organizes investigations focused in 3 elements: animal biology, plant biology, cell and molecular biology, microbial biology, and developmental biology. And in all cases, we're interested in both the basic questions, answering the basic questions of what happens to these systems when they move into the spaceflight environment, but also how those answers impact our ability to design safe and effective spaceflight missions. Next slide. A major theme for the NASA Space Biology Program is open science, and our strategies for enabling open science span a number of resources. There is a biospecimen sharing program, which helps to distribute excess tissues, which are then housed in an open repository, NBISC. Data from spaceflight missions go into a variety of archives, most importantly, the Ames Life Sciences Data Archive, and then GeneLab. Next slide, please. GeneLab is designed to host omics data sets. These are large-scale data sets that you're all very familiar with now. And it now hosts 339 studies. 45 species are represented within GeneLab. We have greater than 10 assays and 130 terabytes of data. Next slide. GeneLab has a number of core functions, including systems to allow the discovery of data sets, systems to allow PIs to contribute new data sets, visualization portals to allow individuals to explore the data in depth, and an analysis portal to allow individuals to run new analysis data as well. Next slide. But what we're here to talk about are GeneLab analysis working groups. So what are these analysis working groups? Well, they began in 2018. They were kicked off at a workshop, and it now consists of 150, more than 150 scientists from multiple space agencies, academic and other institutions from around the globe. These groups meet monthly, and one of their major goals now is to publish peer-reviewed papers using the data available on GeneLab. And you'll have an opportunity to hear about their work today, and also encourage you to join these groups if this work interests you. Next slide. Our working groups are divided into four subgroups: an animal working group, a multi-omic working group, a plant working group, and a microbial working group. Although we have multiple members who span many of these working groups. These are cross-disciplinary groups that interact frequently. Next slide. A new working group is the Ames Life Sciences Data Archive working group, whereas the GeneLab working groups have been focused on omics data. There is other types of data is available within the Ames Life Sciences Data Archive, and we now have a working group dedicated to analyzing this data and publishing work from this data, as well as helping us to develop some data standards. Next slide. So this is a lot of text on here, but this is the charter of the group. We do have a charter. So these groups have some core tasks, which include informing GeneLab on the appropriate data standards, analysis standards, but also we really want to see these groups reuse the GeneLab data. The entire reason for GeneLab's existence is to maximize the utility of these spaceflight experiments. So these groups, we really like to see these groups find data sets of interest, combine data sets of interest, and ultimately publish peer review manuscripts that advance the state of space biology knowledge. Next slide. What are the expectations? Well, the expectations are really the most important thing is contribution. We want people to be energetic, to contribute their domain knowledge, whether that's a certain core data analysis technique or biological knowledge as well. These are open groups, so we expect individuals to participate openly and to be good citizens of these groups. And but above all, it's really about bringing an energy and motivation to these groups. And the benefits, I think, have become quite evident. We'll go to the next slide. These groups have shown themselves to be, oh, I think we missed that slide, to be paper publishing machines. And some of the most impactful papers in the last year have come out of these groups. And so I think there's a real opportunity for all scientists at all levels, whether young scientists or established scientists, to really to publish and to gain new skills and to network, to develop new grants to submit to solicitations and advance their careers, and also to really be involved in spaceflight research, which is a very exciting opportunity. Next slide. So this is our day 2 of our showcase, and again, what we hope to do is really just take a moment to recognize all the hard work of our AWG members, to allow them to show all their great work, and then hopefully this motivates people who are not in our working groups to sign up and to bring their skill sets and their enthusiasm to these groups. And with that, I'll hand it back to Lavorka, and thank you all for your attendance.
Wonderful, thank you, John. So our next speaker will be Amber Paul. Amber is the Embry-Riddle Aeronautical University Wessel Fellow. Dr. Paul received her PhD in molecular biology from the University of Southern Mississippi. She completed her doctoral training at NASA Ames Research Center as a University Space Research Association Fellow and is currently a visiting scholar at Blue Marble Space Institute of Science. Dr. Paul's research focuses on immunity and neuroimmunology endocrine crosstalk in extreme environmental conditions. So, Amber, go ahead and take it away.
Okay, can you hear me?
Yes, we can.
And can you see my screen?
Yes.
Okay, perfect. All right, thank you, Lavorka, for the introduction. As she was saying, my name is Amber Paul, and I'm a new assistant professor at Embry-Riddle Aeronautical University. And before I came here, I worked as a postdoctoral fellow in the USRA NPP postdoctoral position at NASA Ames Research Center. And when I was at NASA, I studied the effects of deep space, simulated deep space conditions on the immune system. So there are a few stressors of spaceflight. Just move this down here. That astronauts will be exposed on deep space missions. And this includes things such as altered gravity fields that would be experienced during launch and landing, so hypergravity forces that are greater than 1G. In flight, microgravity at 0G, and on the lunar and Martian surfaces, astronauts would be exposed to partial gravity. Social isolation, as we know, has substantial behavioral and physiological impacts. What I'm interested in is studying the effects of ionizing radiation on these systems, in particular looking at the effects of galactic cosmic rays, solar particle events, gamma and X-rays. Astronauts will also be exposed to enclosed or hostile environments, so environmental conditions that can induce hypoxic and hypercapnic conditions. The distance from Earth is another stressor that astronauts would be exposed to in terms of pharmaceutical preparation missions and the availability for emergency medicine. And then also circadian shifts can have impacts in terms of the physiological systems that astronauts and humans are not generally used to. And so combined or individually, all of these stressors can impact the immune system. As an overview of the immune system, all leukocytes, they're derived from hematopoietic stem cells that are located in the bone marrow. And these cells can differentiate into myeloid cells or lymphoid cells, with myeloid cells being the precursor to many of your innate immune cells, as well as to red blood cells, and your lymphoid cells being precursors to essentially your adaptive immune system. And these cells will mature, differentiate, and migrate to appropriate sites for function. Myeloid cells and B cells, they mature in the bone marrow, while T cells, they mature and they migrate and mature in the thymus from the bone marrow. Mature cells are then distributed to different parts of the body in terms of, this is called their secondary lymphoid organs. And these include things like your lymph nodes, your spleens, your Peyer's patches in your gut. And these are really sites where activity can occur. So activation of your innate immune system will trigger differentiation, antigen presentation, and have functional consequences. And then post clearance of a certain type of antigen, a certain group of lymphocytes will convert themselves into memory cells. And this is essentially what your adaptive immune system is. So we know that spaceflight can impact different immune cell types at different developmental stages, and we have seen in both short and long-term spaceflight missions, in particular in humans, there is immune system dysfunction. And primarily what's observed is there's this elevated white blood cell count in the form of granulocytes in astronaut blood during flight, and at landing, a very stressful impact on your human physiology. And this goes along with unchanged or reduced lymphocyte counts based on the report that has described these types of populations, and reduced T cell function, and that's been reported by a few different people. We know that viral reactivation in terms of herpes virus reactivation occurs, and that's sort of a function of how well your cytotoxic T cells are working. And there's also been instances of allergic-like reaction in flight as well. We know that the oxidative stress response is induced, and that's where there's an excess production of reactive oxygen species and compared to antioxidants in the system. So there's this redox imbalance. And we know that mitochondrial dysfunction is a key to this process. There's also elevated DNA damage that has been reported in the NASA twin study. There's also mild, persistent chronic inflammation, which is termed inflammagene, which is really seen in the elderly population. So it's this non-resolving mild inflammation. And there's also an increase in the cellular stress response. So elevated heat shock proteins have also been noted in spaceflight as well. So our group identified an elevated granulocyte to lymphocyte ratio and neutrophil to lymphocyte ratio has been detected in flight and at landing. And this has been shown in both rodents and in humans. And an increased neutrophil to lymphocyte ratio, or NLR, and GLR, granulocyte to lymphocyte ratio, are currently used as prognostic measures to measure subclinical inflammation on Earth. And it has been shown to be increased in multiple inflammatory diseases, such as in advanced cancers, in arteriosclerosis, and diabetes, and inflammatory bowel disease. An elevated NLR is also shown to be persistent in prolonged Heinlein loading. So at 14 days of Heinlein loading, we detected elevated neutrophil populations, and that coincided with an elevated neutrophil to lymphocyte ratio in circulation. And this was also shown at 30 days of HU. So at both 14 and 30 days, there was this elevated neutrophil to lymphocyte ratio and actually an increased potential in comparison to an earlier time point. So this persistence of neutrophils in circulation is essentially problematic because neutrophil turnover should technically be short-lived in circulation, and its chronic persistence in circulation does imply inflammation, which can have a consequence to immune system dysfunction. We also saw that in transgenic mice that expressed catalase, which is an antioxidant, actually reversed this phenotype. And this does suggest that then reducing reactive species in the system can reduce neutrophil migration. So this is an avenue for regulation of the immune system. So in deep space flight, apart from altered gravity and isolation, astronauts in deep spaceflight missions will experience cosmic irradiation through the Van Allen belts and on the lunar and Martian surfaces. And we know that cosmic irradiation originates from the sun, the stars, and from supernova. It primarily consists of photons in the form of gamma and X-rays, and in high energy charged particles or alpha beta particles. Galactic cosmic rays are one of the main types of irradiation types that astronauts would be experiencing on a deep space mission, and that consists primarily of protons, helium, electrons, and high energy ions like carbon, oxygen, magnesium, silicon, and iron. And solar particle events is another type of irradiation that consists mainly of protons, and it will fluctuate based to the solar flare calendar. There will also be secondary radiation effects that would be a result of spaceship shielding that astronauts in deep space missions will also need to consider. So total body irradiation with high energy charged particles or protons that astronauts would experience in deep space are not modeled in humans for obvious reasons. So the rodent model is utilized here for ground-based studies. And this is really important to study total body irradiation to these damaging types of particles to understand the impacts on both the immune and non-immune responses. So NASA Space Radiation Labs at Brookhaven National Labs recently identified a simulated GCR and SP dosing scheme that could be used for ground-based testing. So in this table pulled from Simonsen et al., they identified what types of dose equivalents would be, what is experienced on the ISS and low Earth orbit at 1 year is approximately up to 200 millisieverts. And that's in comparison to what we experience on Earth at a very low cosmic irradiation dose of 0.33 millisieverts per year. And they were able to predict that in deep space mission habitats for 1 year, this would be upwards to 650 millisieverts. So this is really a good comparison to see that astronauts would definitely be exposed to higher dose irradiation schemes in deep space. In terms of how cosmic irradiation impacts the immune system, so we know that the immune system are very radiation sensitive cells, and it's typically seen as chromosomal damage in lymphocytes, response to GCR radiation, and this really does depend on the dose of irradiation that's administered. There is this pre-exposure low-dose photon gamma irradiation effect that may decrease lymphocyte regeneration following a second exposure to SPE. So there's this element of radio resistance that could occur. And then whole body ionizing particle irradiation in mice can induce lymphocyte cytotoxicity, and it can impact the function. More recently, Parasite and al. In 2020 identified that low baseline DNA damage correlated with age, a higher immune response post-GCR radiation. So there is this interesting connection between how DNA is damaged and the immune response. Wu and al. In 2020 identified that space radiation in the form of galactic cosmic rays inhibits angiogenesis. And while this is not directly linked to the immune system, this certainly does impact how the immune system will function in terms of its migration patterns. Krukowski and al. This year identified 7 days post GCR radiation of 50 centigrade, identified elevated monocyte counts in blood. And this correlated with defective spatial learning paradigm in mice. So it's interesting to see how this chronic inflammation actually has an impact on other organs, in particular the nervous system here. And Liakis and all this year identified an increase in fatty acid abundance post-irradiation at 4 months in the spleen. And this can actually be really important to consider the fact that fatty acids are important drivers of inflammation. So our group identified that deep space irradiation caused very distinct immune profiles. So for this report here, we acclimated mice to their cages for 3 days. We then Heinlein unloaded them for 14 days. And on day 13, we irradiated them to different types of irradiation, including GCR, SPE, and gamma irradiation. 24 hours after that, we collected their blood and isolated their plasma. And we also did some organ and body weight measurements. And in general, we saw an overall drop in the CD45 population, which is a marker for white blood cells. And the most dramatic effect that we saw were in the T cell populations, which I'll get to in the next slide. We also were able to compare these results to a large cohort of astronauts where we essentially saw mild inflammation as measurement of their IL-1 family. So as mentioned, the main effect that we saw was really shown to be a cause of massive reduction in the T cell population, and that included T helper cell populations in terms of GCR exposure, and T cytotoxic populations in GCR exposure and SPE exposure. What was interesting is that gamma really didn't have an impact on terms of cell number, and even at a very high dose. And we hypothesized this may just be due to the kinetics of cell death pathway, or may not have been as damaging. When we isolated the blood from these animals and we did some qPCR measurements of some pro-inflammatory markers, the main thing that we saw that really grabbed our attention was this high induction of IL-4 across all simulated spaceflight conditions. And IL-4 has really a big role in driving a type of helper cell population called the T helper 2 population. And so we think that maybe in recovery of this result, there may be a higher potential for T helper 2 population development. We also saw an elevated neutrophil to lymphocyte ratio across all simulated spaceflight conditions, which further highlights there is this sort of inflammatory response that is occurring. When we looked at microRNA profiles, we found a large proportion of downregulated immune gene-related pathways, which does suggest overt immune suppression at a higher level. But when we really drove into this a little bit, we saw the specific reduced pathways were really involved in things such as myeloid cell differentiation, which certainly can have an impact on how myeloid cells differentiate and are then activated in response to certain challenges. We saw reduced FCR-mediated signaling, which can impact phagocytic function. And we also saw reduced spleen and thymus development, which could certainly impact how lymphocytes are circulating in the system. We also saw elevated pathways that were involved in primarily humoral immunity, which is essentially your B cell immune response and antibody production. We saw increases in DNA repair and apoptosis being triggered, and we also saw an increase in reactive oxygen species as well. So in closing, there were distinct immune profiles that were identified in each irradiation condition. And really the effects of longitudinal or even sex-specific immunity are really important considerations for future studies. These studies only looked at female mice, and this was only at 24 hours after radiation or 14 days after Heinlein loading. GCR and SP targeted total white blood cell counts, and there was this preference for T cell populations. And gamma radiation, again, did not impact these counts at a very high dose, and that really does highlight the damaging effects of GCR and SP particles in comparison to gamma photons in total body irradiation. There were inflammatory mechanisms that are still important to consider, and how these connect with increased neutrophil to lymphocyte ratio and elevated neutrophils in flight that are some considerations that my lab will be looking into further. And the possible preference for T helper 2 immune cell development in recovery was another pathway that was of interest. And this is sort of relevant in terms of the connection to allergic-like immune responses that are experienced with astronauts in flight, which they experience infrequent rashes and allergies in low Earth orbit. So the connection in terms of this population and how they drive allergic-like immune responses in astronauts is interesting. And collectively, this is only a snapshot into the immune response and response to even a particular set of stimuli. So really understanding how immune dysregulation relates to systems biology is really an open question. And this does highlight the power of GeneLab involvement and omics-based learning to address these kinds of systems biology questions. So with that, I would like to acknowledge everyone at the BNL group, in particular, Dr. Ashley Bethesdy for help with this BNL project, all of my funding agencies, and all of my collaborators that participated in the research that had been presented in this presentation today. I would also like to thank the Animal AWG members for all of their ongoing support, and the GeneLab processing team for all their hard work in generating GeneLab databases. And I would also like to thank the AWG organization committee for inviting me to speak today. And thank you for your attention, and I'll take any questions.
Awesome. Thank you so much, Amber. All right, we have some questions for you here. First one is, will the risks associated with spaceflight be different if traveling to Mars?
So the risks that I mentioned in this presentation were essentially highlighting the fact that these are deep space considered risks. And yes, they would all be considered risks on traveling to Mars.
Wonderful. It is noted that the gut microbiota changes in astronauts and that the gut microbiota and immune system have a strong connection. Could this be a potential reason for immune dysfunction in astronauts?
That's an excellent question. And yes, there is a potential for that. I don't know the degree. I know in terms of the microbiome, it's important to be balanced. So there is that potential of an imbalance that can cause activation through pathogen-associated molecular patterns. Your gut is generally tolerized to our gut microbiome. And in the instance that there is an imbalance in our microbiome could certainly trigger the immune system to go the opposite way than being tolerogenic. And also, I don't think it's just the gut microbiome that's involved in these, because I think in general, there is just a different physiology that's happening in space that may be triggered from events like ionizing radiation damage or other. That essentially set off a cascade of events, and it may not just be pathogen associated, but danger associated. So PAMPs and DAMPs are essentially what trigger your immune response, and I think those are both involved in spaceflight conditions.
Awesome, that's very interesting. Next question is, did you look at Th17 cytokines such as IL-17?
I did not look at Th17 cytokines, but that is certainly something to look further into. I am interested in that Th2 response, more so in particular to the fact that we saw elevated IL-4 with that population of IL-17 being related to Th17 cells. I believe I saw no difference with IL-17 cytokine levels, but did not look at the actual population. So there may be something there to look more into.
All right, I think we are at time, but maybe we can just squeeze in one more question here. Is the radiation exposure dose beyond low Earth orbit estimated based on radiation exposure on the ISS or relative to some other shielded or unshielded environment?
So I'm not sure exactly how they do predict these. I know these are very brilliant scientists at NASA Space Radiation Laboratories, and I'm assuming they're gathering data from all the different sources that are available at their hands, which are including dosimetry on the ISS as well.
Wonderful. Thank you so much for your time, Amber. It's greatly appreciated.
Thank you.
And our next speaker is Christine Meusel-Eichinger. She is a professor at Medical University of Graz. Christine is a full professor at the Medical University of Graz, Austria, and the head of the Center for Microbiome Research. After her PhD in Regensburg, Germany, she joined the NASA Jet Propulsion Laboratory for a one-year postdoctoral period. Since then, she is involved in numerous projects dealing with planetary protection, microbial life in confined spacecraft, and mock-ups in astronaut health. Take it away, Christine.
Thank you very much. I hope you can hear me and can see my screen.
We
yes,
certainly can.
Can you confirm?
Yes, we can.
Okay, thank you. That's good. Yes, I'm talking about microbes in space, relevant environments, and I'm starting with our research questions. So we are mostly interested in learning how human and environmental microbes actually interact during isolation periods with respect to stability and the influencing parameters. We are also interested to understand how we can create safe space travel conditions with respect to infections and health of the astronauts. And we are trying to understand the importance or the interconnection between human spaceflight and problematic planetary protection issues that might come up because of that. And we're using mostly multi-omics and microbiome analysis to address these questions. So as mentioned already, we are looking at ground-based simulations and space environments whenever possible. And I've brought a little bit of a selection of our recent publications which deal with ground-based simulations. One was dealing with Mars 500. Mars 500, as you might know, was a simulation experiment maintained in Moscow. And it took for 500 days, and we used this opportunity to analyze the microbial dynamics within this confined environment. Then we performed a study with respect to the High Seas 4 mission in Hawaii, which was a one-year isolation study. I will show a few more details on that. We had one mission on the International Space Station where we looked at the microbiome dynamics over a 3-month period there. And now we are asking the question how we can create the crewed space flight to Mars, and it's the problematic planetary protection background maybe due to it, and how we can maintain also a healthy human microbiome during such a space flight. I will focus on the two more black parts here, and I will try to explain a bit more what we have done in these experiments. So let me go to the simulation High Seas 4 mission. As I mentioned, it took place in Hawaii. We are looking at these simulations because the real missions are really rare, as you're all aware of. This was an isolation study for 1 year, and it was with 6 crew members. We were interested in understanding the undisturbed interaction of humans and microbes. For that, we gained samples from human skin and also the built environment surfaces. And one of the research questions we had is whether the human skin loses microbes over time. This is a general question with respect to the microbiome in general. So there is the discussion whether when you are in isolation, you're losing certain microbes and you're also losing, of course, specific functions maybe of these microbes as you're not in contact with so many people and the environment. So how did this look like? Basically, the 6 crew members were housed in this dome. It was called dome. It has about 12 meters in diameter, and in the upper floor you see the different sleeping units and the bathroom. On the ground floor there was the kitchen, the main room, and another bathroom, and the lab here. So basically the people were inside all the time. They could also go outside, but they had to wear a spacesuit and they had no contact to any other people. So what did we find, or what did we look at? Basically, we looked at the microbial signatures on the built environment surfaces and the crew surfaces, which is the skin. And we observed that in different phases, we had different important microbial signatures. For instance, as you observe here, Acinetobacter was increased at the latest phase of the isolation study on the built environment surfaces. So each of the phase had a specific indicator organisms. One very nice finding also was that Staphylococcus there was a lot of fluctuation according to this genus, also on the skin. So this was kind of a surprising finding that also on skin it's not stable. What we also could see basically is that there was also some problems with the sanitary facilities, as we could see with Methanobrevibacter signatures on crew skin samples. Basically, Methanobrevibacter is an associated microbe with the human gut system, and we saw this on skin because there were some problem with the compost toilet, and basically, remnants from the compost toilet were spread on the skin of the crew members. So with respect to the stability of the microbiome composition over time, we were also a bit surprised. On the one hand, the built environment was very stable. So these are also the surface samples that we looked at, and you see that the diversity of the microbial community was really, really stable over time. But however, the crew microbial community on skin changed, and diversity increased over time, also being a little bit homogenized with the samples from the surfaces. This was quite interesting because it has not been observed before that skin of person can also adopt microorganisms from the environment, and this is what we could clearly show here. So we could also perform source tracking, so we could observe microbial exchange within the crew members. So each of this chart is one crew member. And if you look at crew member D and F, you would recognize that there obviously was a lot of exchange with respect to the microbiome. And we could also confirm that these crew members had more contact than the others. So what are our conclusions? Basically, we showed that the abiotic surface microbiome remained stable over 1 year, which was really surprising. Also, we saw that the microbiome of the skin was variable and, also more surprising, capable to take up microorganisms from the environment. Overall, we observed a longitudinal homogenization of crew and surfaces with respect to the microbiome. So if you're interested to read more, please refer to the article which appeared in Microbiome Journal a few months ago. So let me jump into our ISS project. It's called Extremophiles, and it was conducted in 2017. We were interested in the microbiome dynamics within the 3-month period, and we picked a period where we did not have any crew exchange, so we were interested in the stable microbiome within the ISS. And this is now Jack Fisher. He is taking samples here at this picture, and he took the samples and they were sent back, and we analyzed them in the laboratory. So what did we find? Also here we found basically, well, it's a bit different to what we found on ground, that the microbiome of surfaces, it was dynamic on the ISS. So over the period of, we saw that there were some fluctuations. However, we could also identify a core microbiome, which you see here in the middle of this chart. That means that basically there's a group of microorganisms that remains really, really stable over time. We also had, for comparison, dust samples from 10 years back, and also there we could identify these core microorganisms. However, we could also identify very specific microbial signatures for all of the different locations within the ISS. For instance, here you see samples from the waste and hygiene compartment, and you see a lot of microbial signatures that are specific to this environment. So the ISS microbiome is indeed an indicator for the functional area. This is something we also know from ground samples. So what we did next, and this was basically different to other studies that had appeared at that time in literature, we then compared our data with ground environments. So we compared our data with human microbiome samples that were received from different projects on ground. And also we compared our data with indoor environments on ground, so like normal bathrooms, kitchen, and living rooms. And you see here the ISS and the human here and the indoor from ground as a comparison. So what did we find? Basically, the International Space Station microbiomes clustered in between. So it was a mixture of the human microbiome and indoor microbiomes that we also find on ground. And when you look here at this cluster of the ISS samples that we observed here, it clusters indeed in between of nose hand samples or bathroom doorknobs. So basically, we see a very nice mixture of normal indoor microbes and, of course, human microorganisms. However, we were also interested in how different these microorganisms behave or which specific traits they have compared to the ground samples. Actually, we had the hypothesis that due to the very difficult situation on board the ISS, they might have higher resistance potential and must be a major threat to the astronauts. But it was the opposite, basically. If you look at different virulence factors, for instance, or stress response, or resistances against antibiotics, we found usually that the ISS was either on a similar level than compared to the human microbiome or the indoor microbiome from ground, or it was even less. So there was no obvious threat for the astronauts that we could detect in the genomes of these microorganisms from ISS. However, is basically that the microorganisms were more adapted towards interaction with metal surfaces. So we found, for instance, cadmium resistance or chromium compounds. And this is, of course, clear because inside of the ISS, you will find a lot of metal surfaces, and we found that these microbes are adapted to these surfaces a lot. This was also confirmed in a cultivation study that we performed with isolates that we also gained from the samples from the ISS. And we could show that the microorganisms attached to surfaces that we provided to them, and they made also biofilms, and they also kind of attacked the surfaces of the different metals and textiles that we provided. So this is a good indication that they really interact with the materials. So we find a quick summary from our studies. Basically, we could conclude that there was no direct concern with respect to the crew health, which was microbiome driven, but we observed biofilm formation and material integrity. There might be an issue. It has been described already in the past that there might be technophilic microorganisms. Also, this problem is known from the Meier station, that the microorganisms form biofilms and then they also attack the materials. So this is something that we have really to look into. So if you're interested in reading more, I refer you to our Nature Communications publication, and there's more details you can read about. So this is basically it from my side. I would like to thank all my team members, and those names that are printed in black are mostly important for the studies I showed just in a minute ago. And I would like to thank for the funding, also for ESA and DLR. And I would also thank my collaboration partners. Thanks a lot for having me and giving me the chance to speak today.
Wonderful. Thank you so much, Christine. We have some questions for you here. And the first one is, what are some of the potential reasons for that oscillatory change observed in Staphylococcus found on the crew's skin?
That's a very good question. I don't have a real answer to that. We just observed that we could not really tell the reason for it, as the behavior of the people was always the same, and the hygiene regulations were also the same. So there obviously is an internal dynamic of the microbiome ongoing, but we don't know any reasons for that. So if anybody has an idea, let me know.
Awesome. Here's another question. Did you also look at the fungi variation on the skin?
That's a very good question as well. No, we did not. That's a pity, actually. Yes, we should have done.
All right, here's some more questions. Have you compared the microbiome before and after?
Yes, it is included in the HI-SEAS mission. Yes, we did. It was also included in the chart that I showed. So basically the first more the left and the last on the right was before and after. And it was still following the trend. So within the 1 month afterwards, we could not show that there is a re-establishment quickly from the skin. So it followed this trend still.
Wonderful. Next question is, do you think there would be dramatic changes in crew microbiomes as new crew members rotate through the ISS, especially from countries with different diets?
That's a very good question. So in these studies, we only looked at the skin microbiome, basically. So I guess you would see the income of a new person, definitely, because based on the source tracking, we definitely saw the impact from the member, everyone. So I would guess would see that. The question is whether the diet would also have an impact on the skin microbiome, which I rather, maybe just a bit, but not much, I guess. And the gut microbiomes, I think there could also be a bit of a mixing, but it would not be so clear as it would be on the skin, I guess. But there is no data. So basically the diet has more impact on the gut microbiome, of course, than towards to the skin.
All right, here's another question for you. They say, amazing work. How do you see your data in comparison with the ISS astronaut exchange published by Venkat and Danko?
Not sure if the question could be a bit more specific.
Whoever asked that question, can you add a little bit more detail,
Maybe
perhaps?
you specify what's exactly your question with respect to the astronaut exchange. I'm not sure what you refer to.
Okay. We will jump until that gets fixed. We will jump to the next question here. Are any of the strains pathogenic?
Yeah, many actually, but this is nothing that is really crazy because everybody of us is carrying pathogenic microorganisms all the time. So they are basically part of our own microbiome, and it's not surprising that we are also having pathogenic microorganisms under isolation or in the International Space Station. Usually our immune system and also the microbes on our skin also and gut really take care of these pathogens, though they do not predominate at some point. However, there was the fear that if the immune system kind of is compromised, pathogenic microorganisms could be on the rise. In our data, we did not see something like that. So there was no concern with respect to that. But of course, pathogens are part of our microbiome.
All right, and the last question here is, rhythms may correlate with periodicity of skin cell proliferation, circadian and circumseptan. I don't know if I pronounced that correctly. I think that is more of a comment than a question.
For the dynamics, I guess, yes.
Yeah, yeah. All right.
I got a comment here, maybe that helps with respect to this interaction. There were some data indeed on the exchange when a new crew member came in, and I think they could indeed track it, as I remember correctly. So I think that might be adding to the question that we could not answer before.
Wonderful. Thank you so much, Christine. That was a really fascinating talk.
Thanks for having me.
All right. All right, our next speaker is Chris Wolverton. He is professor at Ohio Wesleyan University. Chris Wolverton is the Albert M. Austin Professor in the Natural Sciences and Chair of the Department of Biological Sciences at Ohio Wesleyan University, an undergraduate college in Delaware, Ohio, where he teaches courses in introductory cell biology, genetics, and plant physiology. His lab studies plant gravity perception and its influence on the control of differential growth. Take it away, Chris.
All right, great. I will assume everyone can hear me unless I hear otherwise.
Sounds good.
Good. Great. Well, I'm excited to be here and share a little bit about our spaceflight experiment conducted a couple years ago now on ISS. And we're just now getting around to the sequence data that we brought back from the tissues that we were able to bring down from the flight. And before I get into the details of the experiment and our findings, I just want to acknowledge a project like this, a station experiment. A lot of times we can kind of think of it almost as routine, and it's anything but. And so there's a huge collection of collaborators and the NASA centers that are involved in successfully conducting these experiments. Dozens of people at each of these. No room on the slide to include the number of people, but this was also an experiment on the European Modular Cultivation System, which is an ESA facility, and so I want to acknowledge them, as well as the Norwegian User Support Operations Center. My collaborators at Ohio University, and as Lavorka mentioned, I'm at an undergraduate institution, and so I've had somewhere around 20 student collaborators on this project, and I really appreciate each of them as well. So let's turn to the experiment. The kind of experiment, the question we were interested in was one involving gravity perception in plants. We were interested firstly in the minimum amount of gravity that plants could detect. So we can think of that as kind of a threshold question. What's the least amount that's necessary to orient plant growth? And this is where the European Modular Cultivation System and specialized hardware that was developed for that system really allowed us to address this question more precisely than any other instrument or certainly than any ground-based experiment. And that specialized hardware you can see on this slide here involved growing small seedlings of the model organism Arabidopsis in this Kleenex box, kind of a tissue box size piece of hardware. And you can see 5 seed cassettes arrayed on this hardware. And this hardware fits onto a centrifuge rotor, onto an arm of a centrifuge rotor, which has 4 arms and so holds 4 of these devices. This is controllable to initiate germination by hydrating the plant material and by controlling lighting. What we did in our experiment was to grow these plants and trick them into thinking they were experiencing 1G, even though they were in the weightlessness of the space station by spinning the rotor at a RPM equivalent to 1G. And after a particular period of adaptation and grow out, we stopped those rotors and then applied a lower acceleration force. And you can see here the number of treatments we were able to apply that's based on the fact that each of the seed cassettes is a different distance down the rotor arm. And so they represent a different radius and therefore a slightly different fractional gravity level. And this is what allows us to ask this question most precisely: what's the minimum amount of gravity that plants could detect? And so in our experiment, we were interested first in reorienting the plants once they've grown under a simulated 1G to reorient them to the side. As you can see here, this photo on the left is following a 12-hour phototropic stimulation where blue light was delivered to the plants in weightlessness, in microgravity from the left of this photo. And you can see the shoots orienting nicely toward the light and the roots curving away from the light. Those are known responses, and so we took advantage of that as a preparatory step to prepare our seedlings so that then we could ask the question, when we turned the centrifuge rotor back on, which of these seedlings under what gravity level reoriented relative to that acceleration stimulus. And you can see here for an example from the same exact membrane, the same exact seed cassette, that all of these plants are happily reoriented to that artificial gravity stimulation. In this case, these are wild type plants, and this stimulus was about 0.1 or 1/10 of a G. And so you can see nice response, especially in the roots, to that level of gravity stimulus. And so our first set of data, really what we spent quite a bit of time on, was doing image analysis on these root images that or seedling images that we got back to address the question of what is the minimum that plants can detect. And in terms of wild type plants, which are shown on the left, those plants are actually, they align themselves relative to the G vector at the lowest spin rate, the lowest fractional gravity we treated with, which was really in line with theory and sort of extrapolations that other groups have made on the ground using various instruments to make that prediction, but was somewhat surprising that we got as good an orientation across all of our samples as we saw in the wild type. We also flew a mutant in this experiment. The starchless mutant doesn't produce starch and therefore has a reduced gravity responsiveness, but it's still gravity responsive. And so we were interested to know whether that residual gravity responsiveness without the sedimenting starch plastids would have a different threshold, what the threshold would be. And in fact, we found that to be somewhere around 0.2 to 0.3, so requiring quite a bit more G-force to align those starchless mutants than what we saw for the wild type response. And we undertook various methods to quantify this, including root angle measurements, as well as kind of blind scoring of response versus no response. And you can see in this chart here, it really requires up around 0.2 or so or greater to have greater than 50% proportion responding to G, whereas in the wild type plants, we saw that very reproducible response down at the very lowest fractional G treatments. We can express that in different ways. As I mentioned, we both scored and measured angles using image analysis techniques to characterize the responsiveness. What you're seeing here is different treatment groups lumped into the particular spin treatment that they received. And so while we had distinct G levels within each of these that we could separate out, what we found is that there's really no significant difference between, for example, 7 1,000ths and 14 1,000ths of a G in their responsiveness. And so these are kind of, you read these, those that are more vertical here represent those that are more oriented to the gravity vector. Those that are a little less are more random. And this is consistent with that chart I was just showing you. Then to move on to the next phase of the experiment, we received seedlings back from the -80 freezer aboard the ISS and isolated RNA from those seedlings and used a low input specialty kit for preparing our cDNA libraries and did RNA-seq on 12 different G levels of these tissues. So again, this is not your typical spaceflight experiment where you're looking at weightless versus a 1G ground control. We actually had the ability to do a 1G on flight, as well as lots of less than 1G, but not purely weightless treatments. We had the 2 genotypes, and then we dissected our seedlings that we received back, those frozen seedlings, into 4 different tissue domains. So a root tip, a mature root region, a hypocotyl, and a cotyledon. And for those of you plant scientists in the crowd, you'll recognize those are going to have pretty dramatically different expression profiles just on their own because they're developmentally such distinct tissues. But then with 4 biological replicates of each of those, it put us at somewhere right around 400 libraries to sequence. So it's a huge data set. I think Jonathan mentioned earlier, they're in the hundreds of terabytes of data on GeneLab, and very shortly that will be increased by about 3 more terabytes once we get this data set uploaded with all the metadata intact. So we did our sequencing on a NovaSeq 6000, 150 base pair paired end reads, and the usual quality control treatments and adapter trimming. For the data that I'm going to be walking through today, I'm going to stay at the kind of exploratory data analysis level. And so for that, I use Salmon for read mapping and quantification. And then I used an Arabidopsis index, transcriptome index, that takes into account lots of alternatively spliced RNAs called RTD2 quasi. And so let's get into some of the data. Our PCA analysis separates nicely and explains a fair amount of the variance in the read data. This is just throwing all of the 400 libraries at the wall, basically, and you can see that we segregate nicely into cotyledons here for both wild type and the starchless mutant. We have hypocotyls here in the center, and then we have mature root and root tip kind of over here, and this is reassuring. This is saying essentially that these tissues are, they're clustering together nicely by tissue, which is what we would expect. But we want to get at our question, right? We want to know, is there a difference in gene expression that is gravity dependent? Can we look at gravity as a continuous variable, in other words? And so to get at that, we did cluster analysis. This is just showing the 1,000 most variable genes across 205 of our libraries. So this is half of our data set that accounts for the wild type data. And so this is all 4 tissue types. And that's why you see this somewhat interesting striping pattern. What this is accounting for is root tissue and shoot tissue and root tissue and shoot tissue, varying as across our gravity treatments. So at the far left in this figure is our 1G. At the far right is our lowest G level, 4 1,000ths of a G that we sequenced. And so you can see not a ton of detail pops out at us because of this sort of striping pattern. So let's get rid of that by focusing just on the root tip. And here's where things get nice and interesting. Here we've got 1G on the left, 4/10 of a G, and each column here represents a separate library. And we're just asking the question, what are the 1,000 most variable genes, those that show the highest degree of variance across the whole sample set? So each one of these rows is going to correspond to a different gene. And you can see this nice pattern that's produced in variation of expression that appears to be related to G levels, with a kind of a low here in the Mars level G, which is interesting to follow up. When we look at the same analysis with the starchless roots, we see disorder, dysregulation, which is fascinating. This is kind of showing the influence that the presence of starch statoliths and gravity detection has on controlling gene expression. And when you lose that, you really become, the seedling becomes quite disoriented and dysregulated with respect to its control of gene expression, at least in this kind of analysis. If we move on and look at, for example, do some further k-means clustering analysis, we'll see in sort of using 4 different clusters, we see that pattern pop out, especially in our 3rd cluster. And if we look at the kinds of pathways that are enriched in that very distinctive gravity-dependent expression pattern that's shown here, we see a lot of things that others have in fact reported. So nothing particularly novel at this level of detail popping out, but it's, we've got response to abiotic stimulus and to various osmotic reactive oxygen pathway genes are differentially regulated in this analysis. Response to chemicals, hormones in particular, cellular response to chemical stimulus. So what these are saying is that these pathways are highly altered in their expression level as a function of gravity. And so if we dive in a little bit deeper and start looking at differential expression analysis, in this case, using a false discovery rate of 0.1, we find very little, just like we would predict from that larger analysis, the cluster analysis, we find very little distinction between PGM root tips, for example, at the 2 medium levels of gravity. And in fact, no differentially expressed genes pop out at the lowest 2 gravity treatment levels, our category D and category E. We do find a few more up here at the 2 highest treatment levels, but here we're just kind of walking up the gravity treatment level. And for starchless roots, we don't really see differential expression within the root tip across treatment levels until we get up into that higher range. And contrast this with wild type, where we see quite a few differentially expressed genes down between the lowest 2. And this is, again, consistent with what we would expect given the physiological responses we saw to gravity as well. So if we dive in again, that we can express that in terms of a heat map, category B and C. This is just taking, for example, these differentially expressed genes and plotting them in a heat map diagram, 2,500 differentially expressed genes between those two treatment levels in wild type. For our highest two treatment levels in the starchless mutant, a total of 9 differentially expressed genes. And so what we've been working on is ways to follow those up, those differentially expressed gene lists in the lab, building a pipeline for knocking each of those out, especially when the numbers are manageable in the low hundreds or less. The other kind of analysis we're interested in doing that we're just beginning is differential expression analysis between Columbia, our wild type roots, and the starchless root within a particular gravity treatment level. And here you can see the differences in expression for particular genes, even just within that gravity category A, shows lots of distinct, 360 distinct differentially expressed genes between our two genotypes. So, and again, what comes next is all of the interacting effects and really trying to use our data set to pull out the effect of gravity using some of our bystander tissue, the mature root and the cotyledons that are not responsive to gravity as kind of built-in internal controls. So I want to finish up there and leave time for some questions if you have any. Again, I'll just thank our great team of undergraduate students here that have, many of whom have finished, and the great folks at NASA and the NASA funding.
Awesome. Thank you so much, Chris. So we have some questions for you here. Let's see what we can get through in about 3 minutes. Question is, now that EMCS has been retired, do you plan to use some other kind of hardware in future spaceflight experiments?
Yeah, that's a great question. So our experiment was the last experiment to be flown to EMCS. It was 15 or so years old and had put in quite a bit of time on station. And so it was retired. There is a new centrifuge facility, the multivariable platform, the MVP, that's developed by TechShot, that one of my colleagues and good friends, Amara Pereira, and her group will be flying an experiment to as sort of a way to test its ability to do a very similar kind of experiment. TechShot has been great to provide a kind of a growth hardware that's very similar to EMCS. So we hope to be able to follow up some of our results that we found on this initial flight across this continuum. We'll be able to apply that similar continuum once we do the transcriptomic work and have some clearer hypotheses to follow up on.
Wonderful. Here we have some more questions coming in here. Were photoreceptor expression levels altered in your studies?
Yeah, it's a good question. We do find a lot of light regulation pathway genes, including photoreceptor, as well as chlorophyll biosynthesis pathway, general photosynthesis. In fact, one of the largest clusters shows some dysregulate or some altered expression in photosynthesis pathway, which is fascinating. The end of our experiment was entirely in the dark, so we imaged using infrared lights, and so we've got these changes in light expression. Even though the plants were in the dark at that phase, they had seen the light, so they were photomorphogenically developed. But yeah, we definitely see some photosynthesis and photoreceptor related expression changes.
And I think we have time for maybe one more question here. The question is, was the Mars gravity more disturbing for the transcriptome than other lower G levels? It's
a really good question. So what you're looking at here is just a kind of a zoom in on that data set I was showing you, the top 1,000 clustering. And 0.38 G, Mars G, is this kind of lowest. The green represents a reduction or a down regulation. And so we do see that as sort of standing out. What we really need to do is a lot of controls, internal controls, to see if those are related more to gravity or more to the growth status, if there was something odd about those particular samples. It represents really 3 or 4 of our libraries that show that most clearly in the wild type. And so that requires a little bit more massaging, I think, of the data to make a strong conclusion. But it is quite interesting, in fact, that that's right around where we see the activation of the starchless roots become much more gravitropic or gravity sensitive, as well as these apparent changes.
Awesome. Thank you so much, Chris. We are out of time. I appreciate your presentation and your time. All right, our next speaker is Sarah Wallace. She is a microbiologist at the NASA Johnson Space Center, and she received her doctorate in biomedical science from the Microbiology and Immunology Department at the University of Texas Medical Branch. In her current position with NASA Johnson Space Center, Dr. Wallace serves as a technical lead in the microbiology laboratory, which is responsible for mitigating infectious disease risk during human spaceflight. Dr. Wallace also performs work aimed at understanding how the spaceflight environment impacts cellular behavior through investigations using both ground-based modeling and true spaceflight. Take it away, Sarah.
Can you hear me and see my screen?
We sure can.
Great. All right. Thanks very much for the invitation to speak today. I look forward to you hearing about what we've been doing and kind of what we have planned. So I'm going to focus a little bit more on actually doing these experiments on board the space station and some of the capabilities that we've put up there, and we hope others will be using. So just really quickly, I am a microbiologist here at the Johnson Space Center, so I'm going to focus on that. That's my field. Talk to you a little bit about the molecular space age and how that's changing microbiology and how we can do it on board ISS. Why do we want to do these things in space? Talk to you about how we've developed sample prep and sequencing capabilities by walking through a few of my different payloads and then kind of going a little bit beyond micro to some additional capabilities. So first of all, just a quick background. The goal of my lab here at JSC, so we're really responsible for crew health and vehicle health. So we really look at risk associated with microbes in the air, surfaces, and water of ISS. But then we also, as all of you know, microbiology touches everything. So we're involved in all of the programs. And we do a lot of research, a lot of technology development, as well as support all of the different spaceflight programs. And so what I'm really going to focus on today is the new technology capabilities and what we're hoping that becomes kind of a permanent fixture on ISS. So just a quick background why I even got involved in all of this. So as I mentioned, we do a ton of pre-flight monitoring, but then we also have hardware on board for the crew to take periodic samples of the surfaces, the air, and the water. And we've been doing this since the beginning of ISS and using these culture-based methods to really try to understand the microbes that the crew are interacting with on a daily basis. And then we use the data that we get to assess risk. And what we can get from this information right now is kind of a microbial load based on what the crew sees. We don't actually have them count colonies. They compare it to a density chart and then just call down a number or letter, and that gives us an idea of about how many microbes are there. But we have no idea what they are until those samples come back to our laboratory. So that's really where my passion for being able to have some in situ capabilities and near real-time capabilities is very strong, and that's what my team is dedicated to working on. What goes with that is a lot of, so we do have these routine samples that we take. A lot of times we get pictures back like this from the crew, and whether it's some staining on the panels or actual what appears to be some type of biofilm coming out of this water hose, they'll send us these and ask, is it biological? And then how should we treat it? And as you can imagine, just based on these pictures, it's really hard to know, is it something that's actually biological and it's growing, or is it, you know, some kind of chemical precipitate, or is it just dust, all of the above? So we really want to have the capabilities to have the crew be able to look into these more without us just kind of guessing. So over the years, like I said, we've been doing this with our culture-based methods. So that started with Apollo, continued with Shuttle, and continues today with the Commercial Crew Program. But as we get away from ISS and low Earth orbit, we're not going to be able to return those samples to really understand what they are. And having a better understanding of the microbiome of these environments is more important than ever as we start to think about planetary protection and what we're taking with us, and hopefully using the moon really as a test bed for all of this. So really having these capabilities in near real time is something we're, like I said, we're very dedicated to. We've kept a healthy crew, we've kept a healthy vehicle. The culture-based approach has worked really well, but we want to get away from that. So that brings me to 2016. And when I talk about this, I like to call that the molecular space age. That's when we first really started seeing molecular biology happening on board the ISS. So the one that takes the record for the first was mini PCR. This was a Genes in Space experiment. Genes in Space is high school students who submit their ideas, and usually through PCR-based types of ideas. And the first one was a PCR. It was the first molecular biology experiment conducted on board. It was by a high school student, Anna Sophia, who is now in college doing great things. So it's a really great program. For those of you who aren't familiar, I definitely recommend look it up. Then that same year in August, our team flew the MinION from Oxford Nanopore Technologies. We called it the biomolecule sequencer because we didn't want to just be limited to DNA. So that's why we didn't call it a DNA sequencer. And I'll talk very briefly about what we've been doing with direct RNA sequencing on board at the end. But in August of 2016, we demonstrated the first DNA sequencing in space. So before I keep going, why sequence in space? And I think all of you can tell immediately why I'm interested in it. Those microbial questions, what is it? How do we treat it? What should we do? Will become more important as we are, right now we can resupply antibiotics and disinfectant wipes and all the things that we need to treat the water system. But as we move away, we really need, from low Earth orbit, we really need to know when to use those things and when to save them. Obviously, there's huge potential for research, human, animals, microbes, plants, you name it. Medical operations, and then also the way that the MinION works, and I'll talk about it just briefly on the next slide. It's very well suited for potential detection of nucleic acid-based life beyond Earth. So I have a lot of colleagues that are very interested in it from that angle, being able to potentially detect some biomolecule beyond Earth. And the reason that is, for those of you that aren't familiar, it's a nanopore sequencer, and so it's this very small handheld. This is it right up here, the MinION. But it has, in this little small channel, there's thousands of nanopores, actual biological nanopore proteins within a membrane. And you have a buffer that's flowing through there. It's creating you apply a voltage, it creates a current. As you have a DNA or RNA molecule migrating through that nanopore, it blocks the current, and that change in the current is how we get the DNA sequence. So very different than your fluorescence-based Sanger or Illumina platforms you might be familiar with, but works really well, really lends itself well to microgravity. So really quickly, that experiment, we had the same sample prepped on Earth, and we tested at the same time on Earth as the one in flight. It had mixtures of mouse, bacterial, and viral genomic DNA. And what we found through that was that the sequencing on board ISS was really no different than on the ground, and it worked just as well, better than we thought. So that really gave us motivation to keep working. So check out our publication if you want more information on that. So I mentioned that the samples were prepped on Earth, and as you know, that doesn't do us a lot of good to have a sequencer if you can't prep samples in your extreme environment. So at the same time we were flying that, we were working on sample prep, and we were developing a culture-independent method, so we called it swab to sequencer. And we were lucky enough to get to test it out at NEMO twice, 2 summers, 2016 and 2017. And NEMO is a small habitat located off the coast of Key Largo on the bottom of the ocean floor. It's owned by Florida International University, and NASA rents it out for a couple weeks in the summer to send its astronauts down to train. And so the first year, we learned a lot, everything about crew procedures, packing, consumables, all of these things. And so we got some good data. It wasn't great, and we compared everything back to Illumina. But then that second year, we had much improved. We had an improved DNA extraction method, some other things optimized, and we got much better data. And so it was really cool for someone who looks at environmental microbiomes a lot to see impact of the ocean microbiome and start to see a lot of those oceanic microbes in our data set. So again, this is published if you want to go read further about it. So switching back to ISS now. So we got the opportunity opportunity to actually collaborate with Genes in Space. They had mini PCR that I mentioned earlier, and we had the MinION, and we said, hey, let's put these together and do more science. And so this was the only Genes in Space payload, and I think they're on 9 now, that wasn't a student experiment. But now, because we've demonstrated that, students can now use both this whole suite of hardware and procedures we've developed. So what we did was those culture slides I showed you on one of my very first slides. We had Dr. Peggy Whitson was on board at the time, the astronaut. Open up one of those slides in the safety of the microgravity science glove box and select a few colonies to touch and take a few cells into our method. And so when we got the data from flight, she selected 3 colonies. We saw that 2 of them were Staphylococcus we saw 2 IDs, I should say, Staphylococcus hominis and Staphylococcus capitis. We got the plate down just like we normally would, and we ran it through our traditional things that we use, our standard methods here on Earth when we get samples back, which includes both a Sanger sequencer and a Vitek. We do some biochemical analysis. And we found that indeed 2 of those colonies were Staph hominis and 1 was Staph capitis. So I joke for me that this was the best day ever because it was a huge step forward in spaceflight microbiology, because for the first time ever, we identified an unknown microbe off Earth. So this was really exciting. Again, though, we were still dependent on culture, and we were really excited to move beyond that with our swab to sequencer method. So that brings us to the best payload, and that's exactly what we've done, is implement our swab to sequencer. And so you see astronaut Ricky Arnold here swabbing the surface of ISS and then putting that through our method. And we got some great data from these set of experiments. And what we found was that basically, based on what we were sampling, we were seeing the same things we always see from our culture, which is biased to the things that will grow. But the staph, the micrococcus, the bacillus, we were still seeing all of that, but we were seeing things that we would not expect to be able to culture, things that we are sure that are there, and like others have already talked about, normal members of the normal human microbiome. But it was interesting to start to really see them for the first time. Again, knowing they're there, but we don't traditionally culture them. So really just having this expanded view of truly what is there. So again, we've published this part of the data. So please go check that out if you're interested. And we're continuing this work to really better understand the ISS microbiome. So this is really that in situ insight I mentioned earlier. So what we're doing now is called biomole. So it's the exact same thing, that swab to sequencer method, but it's been moved out of a payload and it's moved under to the same group that owns our current culture-based hardware. We call them CHECS, the Crew Healthcare Systems group. And so they're actually running this now. And the goal is really to compare the MinION method with culture-based. And so we've done a ton of this on the ground, and I didn't have time to go into it today, but we've done a lot of this where we take 2 swabs and sample the same area. One goes to our traditional culture-based method, one goes to our molecular method, to really make sure that we're not missing anything, to start doing those comparisons, because we've always based our risk on that culture data. So what we want to see is, can we use this new molecular data to assess risk in the same way? So we're kind of doing that same thing on board ISS, close to anyway, where we're having the crew collect a culture slide, and then in the adjacent area collect one of our swabs and run the method to start building up our data set, and seeing if, we found that on the ground that it compares very well, and we're not missing anything that we would normally see in culture. So we're continuing that on the ISS. Another thing that's coming out of this is, for the length of these experiments, we've been having the data downlinked where we do all the data analysis on the ground. We have a current collaboration right now with IBM and Spaceborne Computer 2, where our data analysis pipeline is actually there. And so we're running data that we're getting from these swab to sequencer experiments through that pipeline and being able to actually get the taxonomic profile out on board without having to send any data back. So this is all happened in the past 5 years. So it's very exciting how quickly we've kind of stepped through this. But this is the kind of information we're really going to need to be able to demonstrate as we move away again, as I mentioned, from that comfort of low Earth orbit. So biomole is something that is just very much in work right now. So I didn't get the data cleared in time to show you, but keep an eye out on that for that. Really quickly, I just wanted to touch on something beyond just microbes. So obviously that's my job and that's what I'm interested in doing, but we really want these techniques to be used by everybody for all of their research related questions. So as part of our best payload, we demonstrated direct RNA sequencing on board. So we sent up native RNA, and we had Ricky Arnold walk through all the steps to prep it and sequence it as we did the same time on Earth. And what we found was, astronauts are really good molecular biologists, and the RNA quality, the data that we got was really good. Another one I would encourage you to check out was the Genes in Space 6 experiment, where we actually flew yeast, transformed it with a plasmid-containing to induce a double-strand break, cultured it to allow that DNA to repair, and then sequenced it to look at DNA strand repair. And this was a high school student's idea that we collaborated on. So again, just using these techniques to do much more than just microbial IDs. So what's next? So as I mentioned, where biomol is happening on board right now, we're continuing to collect that data and look at that comparison. We're doing onboard data analysis. We're working on methods for water analysis, air analysis, and we also have fungal identification ready to go, where we're also identifying fungi. And then, as I mentioned, really the question for me is, how does this data compare to what we've been looking at for years, that historical culture-based data, and can we use that data to assess risk? And then again, just what does this tell us about the microbiome of ISS? So we are planning to use this hardware on board for Gateway for microbial monitoring. And then we also have some upcoming payloads in work to look at full genomes of bacteria, do more epigenetic assessments, and additionally shotgun metagenomics. So a lot more coming and a lot of exciting stuff happening. So with that, I just want to acknowledge my team. It's the four of us who've been kind of working us all the way through. Myself, Sarah Stahl-Rommel, Aaron Burton, and Christian John, and then my team here. Again, Sarah, Hong Win, Christian Castro, Brandon Dunbar, Christian Mena, Tanner Hamilton, and Marie Sharp. And then we've had 8 astronauts now who have done this whole thing. And Kate Rubins twice on both of her expeditions. So many thanks to all the astronauts, all the collaborators, all the flight team. I'm going to need to have 2 acknowledgement slides to fit everybody on here in the future, but just a big thanks to my team who've helped make all this happen. And with that, I will take any questions.
Awesome. Very cool presentation, Sarah. Thank you so much. We have some questions for you here. And the first one is, how do you deal with the high rate of error with the MinION sequencing?
Yeah, so that was definitely an issue early on. So we started working with the MinION in 2015, and we definitely saw that. But you can go check out our paper from 2016 and how we dealt with that. At that time, we were sequencing both strands, so that was helping. And now we're back to just sequencing the one. But we've seen the error rate improve so much from 2015 to now. And so now in our hands, we have about a 97-98% identity to our reference, and we're not seeing, there's still error compared to something like Sanger and Illumina, but it's improved so much, and there's a lot we can do bioinformatically. I won't get into that. My colleagues help us with that, but it's been such an improvement that we're really confident that this is a good way to keep going until something else amazing comes along.
Awesome. Here's the next question is, have you thought about using the system to monitor or measure horizontal gene transfer with microbial populations on board the ISS?
Yeah, that's a really good question. So it's definitely something we've thought about. And I have some collaborators who have experiments in work to look at that. And so I would say I know of several actual payload upcoming and or ground studies using using bioreactors to simulate aspects of microgravity and radiation. So I would say we have not done that in flight yet, but I would say stay tuned. There's several things, both ground-based simulation and actual flight payloads, that should be happening in the next year or so, and hopefully those publications will be out and shed a lot more light on that. That's a huge unknown and definitely something that should be looked at.
Awesome. Here's the next question. To the best of my knowledge, the sequenced data is downlinked to Earth for further analysis. Are there any actual plans to integrate the data analysis in a computing space platform?
Yeah, so that's actually what I mentioned. We're doing that right now. So that's the collaboration I mentioned with IBM and Spaceborne Computer 2. So we have our entire pipeline, including our reference database. We have it all on board ISS right now, and we are in process of we've done it with a Earth-based generated data set. So we're working now to do it with one of those, our actual ISS generated data sets from Biomol, actually do the complete. So it'll be, so the data you're collecting with the MinION are that raw current data, which is doing the process of base calling is the heavy compute part of that. And so we have all of that on board. Like I said, that's that collaboration I mentioned that will take us all the way through that base calling step, all the way to the final piece of providing that taxonomic profile. So that is, like I said, we've done it with a test database, and we're in work to do it with the real database now, or excuse me, the real data set now. So that should be literally any minute. We're waiting for the time to get that to happen on Spaceborne Computer 2, and then we'll be happy to share those results. So yeah, it's a possibility today. We're just working on making it happen.
Awesome. And I think we have time for one more question here. Have you noticed higher divergence of microorganisms in space versus on Earth? And are you also looking into viruses?
So that's a really good question. So I would say that what we're seeing in terms of kind of the diversity of microbes on ISS, it really is similar to a house or any kind of human occupied environment. We see, I think that we haven't done a really directed study to look at, what are the changes that come from changes in diet and hygiene and things like that, and how does that interplay with the environment change. And so that's something that we're working to collect data now to try to understand better. So I would say, just based on our culture data and the small amount of data we have so far, it really just looks like a human occupied environment. But I think that there's certain areas that if we monitor them over time, with molecular methods, we're going to get more insight into that. In terms of viruses, yes, we've just started a collaboration to look at saliva from crew and to look at viral reactivation. I think it was mentioned in a talk in the immunology talk earlier about viral reactivation in the crew. And so there's a few that we can actually detect in saliva. So we're working on a method to be able to detect those, and we'll do some analog testing first, and then hope to be able to use that on board ISS as an indirect indicator of stress or immune stress on the crew. So again, a small set of viruses now, but I think in the future, the possibility is really unlimited with this technology.
Wonderful. Thank you so much. I think we are at time here. I appreciate your time, and that was a really cool presentation. All right, welcome back, everybody. For our next round of speakers, we will have our early career scientists give lightning talks. And because these talks will be 5 minutes in length, we're going to wait until the end of all of those speakers to finish talking before we do have a 5-minute Q&A. So for speakers going forward, may I please ask you to add the name of a speaker when you type in your question so that I know to whom to address the question. All right, so with that, we will have our next speaker, which will be Parul Gupta. Parul is a research associate at Oregon State University, and she received her PhD in India and is a research associate in the Department of Botany and Plant Pathology at Oregon State University. She's a plant molecular biologist with expertise in genomics, transcriptomics, and bioinformatics. Her research focus is on mechanisms regulating plant growth and development under microgravity. So with that, Parul, I'll have you take it away.
Thank you, Lavarka, for the wonderful introduction. So, can you see my screen?
We sure can.
Okay, all right. Hello, everyone. My name is Parul Gupta, and I'm a research assistant in the GeneLab lab at Oregon State University. Today, I'm going to talk about alternative splicing of Arabidopsis transcripts in response to microgravity. So the transcriptome modulation can be studied using two methods, microarray and the RNA-seq. And both the methods give excellent information about baseline and the differential gene expression data, which leads towards the functional validation of genes. So NASA GeneLab data repository contains a large number of spaceflight transcription profiling studies done on model plant Arabidopsis thaliana. In those studies, various Arabidopsis mutant ecotypes have been used to conduct experiments in different hardware and under different conditions like radiation, hypoxia, dark and light, etc. However, differential gene expression analysis provide excellent information about highly expressed or repressed genes under microgravity. We were interested in more in-depth analysis, which includes detection of alternative splicing events, novel isoforms, novel transcribed regions, and detection of link RNAs and small RNAs. For that, we have selected one GeneLab project named GLDS38, and this was the perfect study for us to carry out in-depth analysis because it has both ground control and the flight data. The transcription profiling was done using RNA-seq, and it also has proteomics data. This proteomics data may help us to validate our findings. So we did the genome-wide survey of splicing events in both ground control and the flight dataset, and found a high number of spliced genes and events in a flight dataset. There are more than 2,200 genes that are unique to flight or spliced, and 98 genes that are spliced and differentially expressed as well. We looked for protein coding genes and the non-coding genes as well. So for example, from chromosome 1, 214 genes that are differentially expressed and spliced, and there are a certain number of genes that are unique to flight and ground control, includes link RNAs and the microRNA genes as well. But there are several genes that were not differentially expressed but spliced. For example, ascorbate peroxidase, this gene shows no differential expression but has additional exon skipping event in flight as compared to ground control. Similarly, the other link RNA gene, which also has no significant differential expression but has alternative 3' splicing events in the flight as compared to the ground control. This link RNA gene is antisense to this gene, which is involved in the seed germination process, and this gene also has no significant differential expression. Another example shows extended 3' region. So this gene is an elongation factor which shows alternate 3' splicing site and thereby may have extended 3' region, which is absent in the reference genome. So based on these examples, I say that alternative splicing is a potential driver of changes at the transcript level and may help plant in adapting to a new environment by providing an alternate ORF or adding or omitting a portion to a protein. So in-depth transcriptome analysis may give us a chance to get better insights to understand microgravity stress and also help improving the reference genome annotations. And it is a good idea to develop and integrate the workflows to the other GeneLab projects on various plant and animal species. For this work, I want to thank all plant AWG members, especially Professor JiaRui, Justin, Colin, Richard, and the SQL facility at Oregon State University. Thank you very much, everyone, for your attention.
Awesome. Thank you, Parul. And just as a reminder to everybody, we will do Q&A for all of the lightning talks once everybody has finished talking. And please remember to put the name of the speaker with your question. So our next speaker is Sami Sarenpa. He is a PhD student at SciLife Lab and KTH Royal Institute of Technology. He is a PhD student in the GeneLab, working in Stockholm, Sweden. His research focuses on method development of spatially resolved and single-cell technologies, specifically on non-model organisms like plants, where spatial data is a key aspect in understanding the molecular functions of the tissue. Take it away, Sami.
Great. You should be able to see my slides and hear me, right?
That is correct, yes.
Great. Yeah, hello everyone. My name is Sami Sarenpa. I'm a PhD student at GeneLab, and today I'm talking about multimodal spatial transcriptomics. And what I'm focusing on mainly is plants, but this work can be applied to basically any organism that there is. So what we are studying, we are trying to unveil the spatial host microbe interactions using the method we've developed, spatial metatranscriptomics. And what we want to do with this method is to, when we have for example, this leaf section or leaf here, we want to study where the microbes are located on a leaf surface or inside the leaf, and study how they are distributed there and how does the host plant reacts to these microbes. And for that, we developed this method, and I will give you a short introduction to how does the method works. So if you listened on Wednesday, my supervisor Stefania's talk about spatial transcriptomics, this method follows a similar pattern. So we have these glass slides where we position a tissue section, and each of these areas on the slide surface contain spots, 5,000 spots, and each of these spots contains millions of capture probes. By permeabilizing the RNA molecules from the tissue to the capture probes, and we can basically take all these molecules and prepare libraries and sequence them. Basically, what we do is to bridge the histology, RNA sequencing, and metatranscriptomics. So what do we capture? With this method, we are capturing the probe. We have the probe of interest, and in our case is the bacterial and archaeal 16S, where we have 4 different probes. Then we have host mRNA captured with the poly-T probe. And then we have eukaryotic ITS-18S probes to capture the fungal diversity. And with all these probes, we are able to target a vast majority of the microbes together with the host mRNA. And indeed, we get 3 different readouts, so the bacteria, host, and the fungi. And just to give you an idea what we can do with this method, here is one of proof of concept experiments that we have been doing. In this experiment, we have infected Arabidopsis leaves with the pseudomonas that express M-cherry, fluorescent protein. And we have imaged under fluorescent light these whole leaves, and we recorded the signal. After that, these same leaves went to our experiment, and where we captured the transcripts of the pseudomonas. And if we look at the signal, so you can see the signal here with the wider, more white dots. And if we compare it to what we captured, it looks very similar. And we actually can capture much more than what you can capture from the whole leaf comparing to one tissue section. And you can see this kind of gradient away from this infection site, which is here. And another very cool thing that we can do with this method is that we can look at microbial hotspots. This is one example. So these red areas that you can see here, they are more, they contain more bacteria than you would expect by chance. And these blue ones are cold spots where you see less. And you can generate the same kind of figures for the fungi. And you can also extract some information as networks. And that's what I had for today. And thanks for your attention. And I would like to thank my collaborators and my supervisor, Stefania, and you for listening.
Awesome. Thank you, Sammy. Our next lightning speaker will be Pedro Madrigal. He's at the European Bioinformatics Institute and ESA Space Omics Topical Team. Pedro is an engineer and bioinformatics scientist. In 2015, he earned a PhD in bioinformatics from the Adam Mickiewicz University in Poland. He has developed statistical methods and contributed to the Bioconductor open source project with packages for sequencing data analysis. In 2021, he joined the gene expression team at the European Bioinformatics Institute to work on the Expression Atlas project. Take it away, Pedro.
Thank you very much, and thanks for the honor to speak today here. So I'm going to present a project that has been done in context of the AWG multiomics. So as you all know, antimicrobial resistance is a global health problem currently, and it is predicted that could kill more than cancer by 2050. So it is also an interesting problem in space because of microgravity, radiation, and confinement. So microgravity could influence indeed the growth of bacteria, and radiation can influence the mutation rate. And it has been shown that microbes exposed to microgravity can become more resistant. So this is also important because for the planned long-distance travels, for example, to Mars, when the trip is going to be a few years, there's going to be a compromised ability to treat infections and reduce opportunity for evacuation. So we thought how can we use the microbial data at GeneLab to investigate antimicrobial resistance in space. So we aim to find antimicrobial resistant genes associated to more than 200 pure strains from the MT1 project, and another metagenome samples which were collected in different locations of the ISS and in different times. I'm going to present very briefly today the results from the MT1 project. So this is a project that aims to catalog and characterize all the microorganisms at the ISS to find microbial diversity and how it influences human health and the development of microbial communities. So very briefly explain, it's quite simple to collect. So you need to use wipes in different surfaces, and then they are processed in Earth and subjected to metagenomic sequencing for analysis. So we thought about using a deep neural network model. So this is a simplification of a method published in 2018 called Deep ARG. So our model has more than 4,000 input units here and 4 hidden layers here. But importantly, the output consists of 30 units, which represent 30 different antibiotic classes. And the output of the model gives a probability of how likely is a gene to display antibiotic resistance. So this is a very nice model because it allows to capture antibiotic resistant genes with a sequence that is below the traditional cutoffs that are used for this type of analysis, but that show a highly likely probability of being of displaying antibiotic resistance. So in this heat map here, you can see the analysis for more than 200 isolates of different bacterial species and 30 different antibiotic classes here. And the color represents the ARG. So we can observe diversity, and we decided to because this is a computational prediction, we decided to select the top 2 species that we found, which are Enterobacter bucandensis and Bacillus cereus, for antibiotic resistant testing. So these are basically these assays in which different strains, in which each strain is placed in the context or is together with different antibiotics. And basically, the smaller the inhibitory zone, the higher the resistance of this strain to a specific antibiotic. We can see they can be classified. Each antibiotic has a different cutoff, but this is the result. And very interestingly, we found a very high correlation with the computational prediction of the deep learning model. You can see here that the smaller inhibitory zones correspond to higher values of antibiotic resistance gene counts, and this happens in both species and generally in different strains. We found a inverse correlation and a very good agreement. So just to summarize the three ideas, so we characterized the antimicrobial resistance at the ISS, and we found these two species that potentially could be pathogenic and more resistant to antibiotics. And we validated some of the computational predictions in the lab. And I would like to thank this great team, especially Afshin Behesti and Kasturi Venkateswaran, and also the Spaceomics topical team funded by the European Space Agency for all the support provided. Thank you very much.
Awesome. Thank you so much, Pedro. Our next speaker will be Henry Cope. Henry is a PhD student at the University of Medicine, School of Medicine, University of Nottingham, and was a NASA GeneLab 2021 summer intern. His research involves applying computational methods within the domains of bioinformatics and computer vision to data associated with the physiological deconditioning induced by spaceflight. Henry is also looking to advance spaceflight life sciences research by writing software for biomedical CubeSat missions and investigating ethical issues surrounding astronaut health data. Take it away, Henry.
Thank you very much for the introduction. So this project is a multi-mission transcriptomic analysis of spaceflight skin. And I've been supervised by Afshin on this project, as well as Nate and Craig. I think I'll skip over this because it's been discussed quite a lot during the workshop in terms of spaceflight having a unique environment which has stresses that can lead to health issues. For anyone kind of new to the field, I would recommend this paper, which is Fundamental Biological Features of Spaceflight. And it's a nice overview of the field. So why study skin for spaceflight? For one reason, skin issues such as rashes and cuts are a commonly reported in-flight clinical symptom by astronauts, and that kind of dates back to kind of shuttle kind of era missions. Skin also has the potential to tell you more about the underlying health of the body, such as the functioning of the immune system, which was mentioned earlier as a key factor of spaceflight health. And that's, of course, well established through kind of dermatology on Earth. And then also importantly for spaceflight, skin is the first line of defense against radiation and pathogens. And so if the skin barrier is compromised, then you get maybe increased risk of kind of skin cancer due to the ionizing radiation. And some of that, like the pathogen stuff, relates back to some of the ongoing kind of microbiome studies and ones that have been conducted in the past on the ISS. Also for spaceflight, skin is easily accessible. It can be monitored using non-invasive methods. So it could be monitored using swabs, and then also hair follicles can be extracted and looked at. And I think it's been understudied in spaceflight to date. So there's been a couple of rodent studies, and so one of them found a skin atrophy of about 15% in the rodents that were flown to space. There's also been astronaut studies that are similar, a bit of a mixed result on the potential for skin atrophy there. And then also there's been the microbiome studies that show similarity to the ISS environment. So in this study, we looked at 5 different GeneLab data, transcript data sets. And because they're from 3 different missions, there's lots of different variables. So different skin tissue types, different diets, different strains, male versus female. And you can see, for example, here the second row and the fourth row from the same mission and the same diet, and that's a slightly modified diet. So it's I think I've lost my cursor. There we go. So that's got an added prebiotic, and then the first and the third row, they have a standard kind of diet, and so you can see from the global kind of expression that these differences pop up. And then all of these samples on the PCA between spaceflight and ground control for all of the data sets, they cluster quite well, which is nice to check. So I don't expect you to read all of this, but the pathway analysis that we've done between the data sets, we find kind of responses that are shared across a lot of the data sets and some that are discrete between different data sets. So as an example, collagen-related pathways are particularly significantly downregulated in the JAXA data sets. Then in the others, they're significant, but they're upregulated. And then keratin is downregulated across a lot of the data sets. So this has led, this pathway analysis has led to network analysis, but we're still kind of refining, but some key hubs start to emerge. A lot of these are, as you'd expect, like collagen, extracellular matrix, keratin, DNA damage, and also mitochondrial. So it's all related to kind of things you'd expect in spaceflight. And now we're in the stage of further investigating these key hubs through, so this is kind of more hypothesis driven. So we've identified the hubs from the data, and then we're looking at the expression of different genes within those hubs. So I'm creating curated lists for things like DNA damage, and then I'll be producing visualizations of those. An example here is with the collagen related genes. It's very clear that the JAXA missions are different to the rodent research mission, possibly due to the sex difference there or the housing differences. And then hair keratin show downregulation, which has been found in previous studies. And then it's a bit more mixed bag for the epithelial. So thank you very much for listening. There's lots of ongoing stuff. So we're looking at astronaut data, but I can't show any of that due to ethical issues. We can look at potential countermeasures. We're collaborating with the GeneLab for High School program, which will be mentioned by Sigrid later. And all of this should lead to a publication, hopefully around December or January. Thank you very much.
Awesome. Thank you so much, Henry. Great work. Our next speaker is Alex Dong-Hyun Siu. He's a research intern at University of Wisconsin-Madison. He's a biostatistics researcher intern at JiaRui Lab who studies statistics and mathematics for his bachelor's degree at the University of Wisconsin-Madison. He will start his master's degree in computer science at the University of Southern California starting in the following semester. His research interests include interdisciplinary data science, Bayesian statistics, and application of deep learning methods. Take it away, Alex.
Thanks, Aborka. Can you hear me and see my screen?
Yes.
All right. Hi, everyone. In this lightning talk, I will present about the work that me and my colleague have done with GeneLab data GLDS 120, the CARA experiment, which is about physiological adaptation of plants to spaceflight. To begin with, I will briefly elaborate about the CARA experiment and the data that we use for our analysis. CARA experiment, short for characterizing Arabidopsis root attraction, is an experiment that was designed to compare the spaceflight transcriptome responses between different genotypes of Arabidopsis thaliana's root tips under ambient light setting. 3 different genotypes were used, which were wild-type Columbia, WS, and 5D mutant of Columbia 0. These plants were grown in ISS for 11 days and was also grown at the Kennedy Space Center for its corresponding ground controls. Then using the processed RNA-seq transcriptome data based on the experiment, the original authors studied about how the gene expression varies by plant genotype in response to the spaceflight environment and impact physiological adaptation differently. They suggested genetic manipulation could be plausible for better adaptation of plant growth in spaceflight, and concluded that the adaptive response is dependent upon the genotype of the plants and the specific environment within which it was grown. Building onto their results and considering this CARA experiment provide rich GeneLab pre-processed data set of different genotypes, we decided to build our own analysis pipeline that starts with outlier detection and ends with clustering to provide new perspective to this experiment. To start with, we applied the most well-used statistical methods in the multi-omic studies, PCA for dimension reduction and K-means for clustering. While simplest method could give you promising results, however, in our case, one cluster consisted 95% of the data for all instances. Therefore, we used this result as the outlier detection and took this one condensed cluster here with red circle to the next step of our pattern recognition pipeline. Next, while we want to proceed with same process of dimension reduction and clustering, this time we consider a different approach. We understand that while PCA is popularly used for its simplicity, it has its limits for losing essential features through embedding, which is often degrades the clustering quality. Alternatively, representation learning is a technique to capture contextual information from complex and higher level features through nonlinear mappings. For the main model, we considered a deep learning-based approach, specifically sparse autoencoder, the model that we used in 3-layer neural network with feed-forward architecture with a sparsity-inducing penalty, which allows us to achieve latent representation in the data with limited data set and less computational cost. The end product here will be encoded data representation at the final bottleneck layer. We believe better data representation deserve better clustering. So therefore, we applied clustering method that could capture the correlation and dependencies between different attributes in the data. Gaussian mixture model with expectation maximization algorithm is the main model that we considered for our pipeline, which will help us detecting the true patterns of the data while also considering uncertainty of a given cluster. The result from our pattern recognition pipeline confirmed the prior belief that spaceflight environment causes stress response from plants, but with more details. We found out the spaceflight stress response is related to oxidative stresses, hypoxia, and imbalance of reactive oxygen species. We used multiple bioinformatics tools to learn more about these clusters, such as their related hormonal pathways and predicted transcriptional factors around identified clusters. This revealed that the environmental stress response would initiate crosstalk between hormonal pathways to cope with their current condition. We also have found that the crosstalk between hormone pathways were different depending on the genotypes, which agrees with prior studies and explained the different phenotypes between genotypes. For example, Columbia 0 showed the decreased root length when growing in orbit with light setting, while Phyte mutant's root length was not affected in the orbit, but rather its diameter and volume is increased. Our result tries to explain this by making a connection with abscisic acid being more relevant for Columbia 0 to inhibit the cell elongation. And as for Phyte mutant, gibberellin acid, and jasmonic acid, crosstalk being more relevant along with the phytochromes regulating the oxygen transport. Finally, with gene ontology analysis and by looking at the differential expressed genes, we predict that this genotype-specific hormone pathway regulate and will contribute to the cell wall remodeling. That's all for this presentation, and thank you.
Thank you, Alex. And our last speaker for this lightning talk series is Eric Hanshin. He is an evolutionary biologist and scientist at Los Alamos National Lab, working on a variety of biological problems, including bioremediation and acclimatization of Arabidopsis thaliana to the spaceflight environment. Take it away, Eric. Eric, have you been able to join? Eric, are you...
And I've promoted him to present, but let me ask to unmute.
Is he at the workshop?
Yep, can you hear me now?
Yes, we can.
And can you see my slides?
Not at the moment.
Uh-oh, this is the problem. Share screen. Share screen. Oh, great.
Let me pull up your slides here real quick. Did you put them in the folder by any chance?
Yes, I did.
Okay, give me a second to pull them up.
Oh, thank you.
All right, I will be sharing right now.
Thank you. So this is a project looking at the alternative transcription response to spaceflight in Arabidopsis thaliana. This is a large collaboration that is NASA funded, primarily led by the University of Washington, but with collaborators at Los Alamos National Lab. Next slide, please. So for this project, we've grown Arabidopsis thaliana in the advanced plant habitat. This is a miniature fridge-sized growth chamber you can see there on the bottom right, which contains about 1 kilogram of arsenite as a soil, as well as some water. And then we've seeded in numerous plants, about 24 plants in each science carrier. And we've grown these out for 6 weeks while monitoring temperature, light, CO2, O2, as well as humidity. And then at the end of those 6 weeks, we have 2 different tissues, leaves and stems, that we've harvested and prepared for transcriptomic, proteomic, as well as metabolomic analyses. And we've repeated this entire experiment twice, so we have 2 entire different grow outs of approximately 6 to 8 weeks long. On the next slide, please. And then this is a side project that I've been working on, looking at alternative splicing in these samples. So rather than going into all the transcriptomics and proteomics today, we're going to be looking at the alternative splicing as a really important environmental stress response pathway in order to deal with the stresses of spaceflight. And for this project, we've used the PacBio Sequel2 sequencing instrument to sequence entire full-length reads. So we're not computationally predicting exon-exon junctions or splice sites. We have the entire transcripts as our raw reads. The next slide, please. So as a broad-scale overview, we're seeing a substantial amount of alternative splicing that is unique to spaceflight. So these are Venn diagrams of genes that are experiencing alternative splicing in any 3 of my 4 replicates for each condition, whether that's ground or flight, leaves or stem. And you can see that across both of our experiments, we see multiple hundred into the low thousands of genes that are unique to flight conditions. I've also looked at all of these overlapping sections, the sections that are just unique to stems, just unique to leaves, and unique to all of these things. And these seem to be genes that we expect, whether they're photosynthesis or cell wall functions and lignin deposition for stems. That just kind of supports the hypothesis that these genes are about doing what we would expect them to be doing, whether they are in leaves or stems. On the next slide, we are looking at the function of some of these genes. So on the top right is a GO-term enrichment for the first experiment. There are substantially more genes that are unique to spaceflight on the second experiment in the bottom right. But when we look at the intersection of these 2 is where things get really interesting. We're looking at hormones that are related to microgravity as well as gravitrophism, ethylene, salicylic acid. As well, we also see that abscisic acid, which we know plays a role in developmental gravitrophism, seems to be alternatively spliced in both experiments 1 and 2 in the bottom left there. On the next slide, the other interesting thing is that when I compare differential expression to alternative splicing, look at the genes that are significantly upregulated, they seem to have a really similar functional pathways. A lot of cell signaling pathways, including salicylic acid, abscisic acid, as well as jasmonic acid. So not only there are some similar genes, but definitely the similar pathways in terms of functional enrichments are both differentially expressed as well as alternatively spliced. But this overlap is relatively minimal, suggesting that alternative splicing is a major alternative response to differential expression in response to spaceflight conditions. This is obviously just a quick overview. We have a lot more data and a lot more depth of this, so please stay tuned. On the last slide, I'd like to thank a lot of my collaborators. There's obviously a very long list of them, as well as some technicians, as well as my funding sources, especially CSES in Los Alamos. And if you'd like to contact me with any questions, there's my email. Thank you very much.
Awesome. Thank you so much, Eric. And unfortunately, we are out of time for Q&A. We do have to move on to the next speaker. I do apologize for that. But our next speaker is Parag Vaishampayan. He is a space biology portfolio scientist at NASA Ames Research Center, and he provides overall scientific leadership and direction to maintain the scientific integrity of projects within the Ames Research Center's space biology portfolio. He supports over 70 active space biology projects to better understand how spaceflight affects living systems in simulated ground-based experiments, the International Station, and preparing future human exploration missions beyond low Earth orbit. So with that, Parag, you can go ahead and take it away.
Thank you, Lorca. Did you see my screen?
We can, but you are not. Yep, perfect. Yes.
Okay. Thank you so much. Yeah, so thank you, GeneLab and the workshop organizers for giving me an opportunity to come here and talk about the space biology funding opportunities. As Lavarka mentioned, my name is Parag Vaishampayan. I'm the space biology portfolio scientist. So basically, with the portfolio manager, we manage all the flight as well as ground projects. So it's very important that we advertise, talk about the funding opportunities to bring the more science. So today I'll be only talking about the space biology opportunities. If you have any questions, we can take the questions later. But before we jump into the actual opportunities, just want to give you, I know it's kind of a repetition, but some of the key elements, the environmental factors which encountered in the space flights are very important. And as a disclaimer, these slides, I borrowed some of these slides from Dr. Sharmila Bhattacharya. She's a space biology program scientist. So really thank you, Sharmila, for allowing me to share your slides. I don't have to go through each factors because some of the speakers have talked about one or multiple factors, but our philosophy is to look into all the factors together and the combined effect. And my next slide is a great transition into that. Some of you might have heard about tides, and tides represent thriving in deep space. This is a key area where space biology or biological and physical sciences, BPS, is focusing from the space biology perspective. So all the future solicitations you will see, they are tied to tides. And so let's understand what tides is, because that will help you formulate your proposals and hopefully win the proposals next year. Tides is basically, if you can see different deep space stresses. We already looked into it: gravity, radiation, temperature, isolation, circadian rhythms. Along with that, to understand that, we have multiple platforms, and the basic one are the ground studies. Then you can go into the low Earth orbit, and eventually we will have a gateway, which is a smaller version of the ISS around the moon. Then we will land on the lunar surface, so there will be missions performing research on the lunar surface, and that's our step for moving towards Mars. So there will be a Mars transit, and eventually humans will land on Mars and perform experiments on the Martian surface. So that's a gradual progression of the platform usage, and accordingly we will have solicitations and funding opportunities coming in. So without going in too much detail, and I'm open for sharing my slides. So if you want to go through the tides, you can please have a look at the slides later on. But there will be model organisms which we will be using. Currently, we are using both unicellular as well as vertebrate and invertebrate. There is a hold on rodent research on ISS, but right now all the calls, and I'll go into that detail, are on invertebrate research on ISS. But as you can see, the progression of the platforms, there is also progression in the model organisms we will be using. So you should take a note of the tides, specifically if you're interested in applying for the next space biology ground and flight calls. So I want to throw this one slide here. Thanks to Sharmila again. So what are the types of the space biology funding opportunities? There is an annual ground and flight research announcement, which recently we announced 10 successful flight and ground opportunities. I'll show a slide later. We have few special opportunities, and I'll talk about that. And definitely there will be, there is an international life science research announcement coming up in the future. For more educational research purpose or research with underprivileged, underrepresented states, there is a well-established program currently funded by multiple agencies, including NASA. It's called EPSCoR program, and I have a slide for that, so I'll talk about that in general. So basically, these are key 4 funding opportunities we have, and I'll talk about each in detail as much as I can. This was our last funding call, and as you can see in that, we have 3 sections. One is animal biology, which is comprised of vertebrate and invertebrate research. Then we have plant biology and microbial ecosystem. The microbial ecosystems actually overlap between plant and biology, because as you can imagine, microbes do share the common environment, not only the surfaces, but also humans, plants, and animals. So they live within and inside. So microbial ecosystem research is very important for both plant and invertebrate research. If you dip down into and see, and this is a table which is there in the call. As you can see, there are 2 main sections. One is ISS flight and /flight/ground experiments, and there is an entirely only ground-based experiments. So in ISS flight, if you see, there are components. One is a research investigation and an expanded integrated investigation. So currently, we are only soliciting invertebrate research. We're not soliciting any rodent research. And in ground studies, which is something more important for this particular group, is the GeneLab analysis investigation. So I know the amount is relatively very small, so 50K, and we intend to fund at least one, more than one also, but at least one. If you have any research proposal, if you are part of the GeneLab analysis working group and would like to propose something, there is an opportunity for here. As I would like to mention here, if you have any community feedback, if you have already applied for the funding, and if you have any questions, the frequently asked questions section in the grant, it's very enriched. We make sure that all your community feedbacks and questions are answered in the FAQs. If you still have any questions, please reach out to Sharmila Bhattacharya, or if you want to talk to us, but the best mode is to talk to Sharmila, because we will compile all your questions in the FAQs. If you still have any questions, please let us know. If you have comments, how to improve the calls, I'm always open to listen and incorporate. So you'll see some changes in the next year, but this was our current call, and we have funded 10 projects. I'll talk about that. A very special, very interesting funding opportunity we had this year. It's called Lunar Exploration Instrument for Space Biology Applications, or known as LEA solicitation. We were expecting at least 1 or 2 proposals we should be able to fund. I'm happy to announce that we have funded 3 proposals this year, and LEA solicitation is mainly, the purpose of the LEA solicitation is to create a biosensor hardware-based with yeast as a model organism, conduct and support the flight ground preparedness experiments, which will be eventually launched on lunar mission. So this LEA solicitation does not cover the launch. This is basically to prepare the ground experimental part. So these are the recently funded PIs I mentioned. So first one was E12 ground flight call, and the second one was E10 LEA solicitation. So under LEA, we have 3 PIs and we have 10 PIs under the E12 call. There should be next call coming out pretty soon. We haven't announced that yet. The next one I would like to talk about is a very special opportunity. I believe I'm running out of time, but I'll quickly cover. Multiple agencies, including National Cancer Institute, FDA, and NASA, they came together and come up with a research topic for extended longevity for 3D tissues and microphysiology or tissue chip, organ-on-chip systems for modeling the acute and chronic exposure to the stresses. So we announced that in June 2021. Selection should be announced in early 2022. Last but not the least, we do have a NASA-funded EPSCoR. Under that, you have, if you belong to one of these states, please reach out to your EPSCoR representative from your institution or go to the EPSCoR site and see if you can come up with a funding call. This was the one we announced last year, solicitation for 1-year ground-based study in the plant or crop research stress. And the second one we had was a CAN on EPSCoR CAN on organismal biology, which was released early this year. It's a 3-year ground-based study, close to 750K funding. We will be announcing the space biology topic CAN early next year, and late this year we will be announcing the rapid research response for EPSCoR. If you would like to start, and if you're not sure where to start, I think the NASA taskbook is the best place where you can start and look for what research space biology and HRP or Human Research Program has already funded. If you don't know the taskbook site, I can copy it here, or the GeneLab folks can provide the link for you, but that's the best place to start. If you have any questions about your prior experience with the solicitations, funding, or if you have any suggestions for the future, please reach out to me. You can look me up on my webpage on NASA Ames, or my email. I can type my email in the chat. But really would like to thank the GeneLab for organizing this workshop. Most importantly, the GeneLab community as such for doing all this great research and producing the data. And last but not the least, the analysis working group is the powerhouse for repurposing the data and bringing more science out. So kudos to all the 3 and 4, 5 now analysis working groups. If space biology as a program can help you anyway, please let me know. I'd be more than happy to take your comments and make any changes for the future solicitation calls. So that's it from me for today. But if you have any questions, I'd be more than happy to take the questions.
Awesome. Thank you, Parag. Let's move on to Sigrid for our next talk. And hopefully at the end, there'll be a little bit of time left over for questions. So Sigrid Reinsch is the Director of Education and Outreach for the Space Biology Program at NASA Ames Research Center. She received her doctorate in cell biology from the University of California, San Francisco, and her postdoctoral training at the European Molecular Biology Laboratory in Heidelberg, Germany, prior to joining NASA in 1998. Sigrid is the Director of Education and Outreach for the NASA Ames Space Biology Program and directs 2 impactful summer internship programs, the GeneLab for High School and the Space Life Sciences Training Program. And with that, go ahead and take it away, Sigrid.
Are you able to see my slides?
Yes.
Are you able to hear me? Okay, yes. You can see. Okay. So yeah, I'm going to talk about GeneLab for high school and GeneLab for university, as well as a few other things. So as you know, GeneLab data democratization is a huge effort. Starting with the data scientists and the analysis working groups, and now we have education working groups as well. And so the topics I'm going to hit on here are GeneLab for high school, GeneLab for university, internships, the SSCP program briefly, and then the NPP program and the STAR program I just want to mention. So GeneLab for high school has been going on for 5 years. It's was started by Elizabeth Glaber, and basically it's a training, a very intense 1 month long training program for about 15 students each year, and now we also have it for teachers as well. And they learn about space biology. So as I said, Liz Glaber is the person who developed the program. She's an absolutely amazing teacher. She's led it for all 5 years. And it basically has 3 core components. They have learning modules where they really learn how to analyze gene expression data. It started out with microarray data, and now for the last 2 years they've used the GeneLab Galaxy platform to learn about transcriptomics and RNA-seq. They get networking opportunities, and then they have one of the main components of it is this research training project where they actually analyze in depth the data set, and working as a team, come up with a hypothesis, and they have to actually propose an experiment to follow on with it. So this is the team for this year that basically ran the program. So Liz and Sam, also Rachel Gilbert, who is a postdoc at NASA and has now moved on, and Kim Cadmus. So as you can see, that this is just data from the number of applicants that we've gotten each year, and so we've increased the number of applicants each year. The demographics, we get a lot of especially Asian students, and that's actually for a lot of our programs that's the largest block of students, and we end up with a pretty good demographic of the students, a really good mix. We really try to target getting equal mix of male and female students, and targeting different opportunity levels as well. It's an absolutely rigorous program. So this is in 2020 we had to move the program to virtual, and that's when I started actually as the director of the program. So it was a big pivot to going from in-person at Ames to going virtual. So in the in-person programs, we really were only able, because we don't provide housing, we do provide them with a small stipend. The students were really only from local schools. And so going virtually actually allowed us to expand to a little more diversity. So we had 3 out-of-state students and even a student from Germany in 2020. And in 2021, we had 7 out-of-state students, and we even had a student from the Netherlands, and only 4 students from the Bay Area this year. So, what is it? What are the components of it? So there's learning modules where they learn about space biology and model organisms, and introduce GeneLab and the analysis platform. They get a lot of networking opportunities with a lot of wonderful lectures by space biologists throughout the country, and even I think internationally. We have networking, and they get some field trips, and then in the independent learning project, that's where they really analyze. They go down, they actually analyze a GeneLab data set, they develop a hypothesis, and then propose research. And I'm going to skip these just for brevity, but they have, it's a really intense program where they really get to do a deep dive into space biology and really learn some really important critical biological principles. And so one of the things that happened this year was that, as Henry just mentioned, Henry Cope, that they one of the projects over the last 2 years was the skin project, and so a number of the students actually focused on skin for their research, and so they are now collaborating with Henry and Afshin on actually writing this multi-skin paper. These are the students that are involved in that. And then we also have a teacher training program. And so how has that worked? And why do we want a teacher training program? Is because we can inspire more students, we can share the resources that we've developed and get them involved also in citizen science. And so Jennifer Claudio has been a teacher in the program from day one. In 2020, I recruited 3 other teachers who participated in the program. Carolina Levy has actually become one of our GeneLab. She's in several of the analysis working groups, and Lori Waters is in the plant analysis working group now. So this is really a great thing. And in 2020, what we did was the outcomes of that was that Carolina Levy taught the GeneLab for High School module at Southern Oregon University, where she was a new professor there. And their students actually helped us with kind of working on the GeneLab Galaxy platform, and also our GeneLab curriculum manual provided a lot of feedback on our new RNA-seq platform. And one of the feedback that we got from this group, what we do is we have an education working group where we meet with the teachers throughout the school year to hear how they're doing with implementing the GeneLab for High School program in their classrooms, is that they really needed time during the course to actually prepare for their own classroom situation. And so they proposed that the teachers should have their own team for analysis, whereas, in the first year when we did this, the teachers were kind of in the little breakout groups for the students. And that the teachers should develop their own curriculum modules for implementing in their own classrooms. And so in 2021, that's what we did. We brought in 3 new teachers, Catherine Boileau, Monique Salazar, and Jennifer Callison-Bliss. And so what they did is they participated in the first couple of weeks as normal in the classroom, where they really learned the steps involved in analyzing. And then for the second half of the course, they go into their own breakout group, and they work on, they can analyze data, but then they also develop curriculum modules to use in their own classrooms. And so that's what was done this year. I'm going to go through this really quickly. This is kind of what it looked like, and they actually had to do presentations at the end. So everybody we have a big symposium at the end where everybody presents their results. And so these are now being tested in their classrooms. And so they came up with 3 modules. They're sharing them between them and trying them each of them in their own classrooms, and the students, and giving us feedback on it. So basically, we get really high feedback on this course. The students love the course. They love Liz as a teacher. She's absolutely amazing. I also wanted to give a big shout out. I realized I forgot a slide of Sarah Wyatt, who also helped provide plant expertise for this. Oops, going a little too fast. Anyway, so it's an amazing course. Our applications for the students are going to open on January 1st, and Sam is going to provide some links in the chat. So GeneLab for University, this was started by Amanda Saravia Butler and Lauren Sanders. Basically, GeneLab has all these standard data processing pipelines, and we wanted to also have students that could help us work on all aspects of GeneLab. These standard data processing pipelines actually were helped. They were developed by our GeneLab analysis working groups. A group of interns came in 2018, I think, and they helped us actually vet these pipelines and get them into implementation. And one result of this is now that Amanda and Lauren and Phil Heller at San Jose State University have developed a really great course that is called GeneLab for Universities, where they really get command line level training in bioinformatics. So it's a boot camp. It's a week-long course. Amanda has developed all of the, wow, I'm getting lots of those. Okay, they have to, let's see. Okay, I'm going to go to the punchline on this, which is that everything is on the GitHub. So this amazing course, which is a week-long course, is now available to educators everywhere. They can teach it in their own universities. All of the lectures, all of the Jupyter notebooks, everything is available online. And it's going to be, I know that Embry-Riddle University is planning on implementing it, and GeneLab is actually hoping to run another boot camp in 2022. And that's being developed in collaboration with the minority universities. That's on here, sorry. And the big problem is actually just getting the compute time to be able to do this, as well as the personnel to run the course, et cetera. So I'm not going to talk about SSTP that much, except that that's another program that I run. The applications for that are open right now. It's a wonderful training program. Everything run virtually. We have mentors, individual mentors, and those programs the applications for that are open right now for undergraduate students. The NPP program is, this is another link for NASA postdoctoral fellowships with NASA PIs. And then the NASA STAR program is for training PhD level scientists who are interested in learning more about space biology. Applications for that open in the spring. And all this funding from the space biology program. Thank you.
Both Parag and Sigrid. We are running a little bit behind, so we do have to cut our Q&A here and move on to our next speaker. So our next speaker is Siva Vanapalli. He's a professor at Texas Tech University in the Chemical Engineering Department and the Brian Pierce Bagley Regents Chair. He obtained his PhD from the University of Michigan, and his research interests are in the areas of microfluidics, complex fluids, cancer, healthy aging, and technology development. His laboratory has been involved in several spaceflight projects with C. elegans, focusing on muscle strength and aging and gut microbiome. Go ahead and take it away, Siva.
Sure. Well, I appreciate the invitation here. And my screen is visible on your side?
Sure is.
Okay. Hi, everyone. Today, I would like to discuss about our project on measuring the strength of this. It's a project on measuring the strength of this worm, and this project is basically micro 16, and it was launched on NG15. So initially, I want to go into the study design of this project and also explain a bit about how we accomplished all these experiments. So I'm not going to show as much data because right now we are in the process of analyzing all the data. So essentially, the overall emphasis is on the design of the experiment and the details in terms of engineering the hardware, which allows us to measure the strength of the worms. So with that, I would like to thank all the team. Often these spaceflight projects involves, different expertise and different roles as well. So I appreciate the entire team, which allowed us to run this experiment on the station. As you all know, in terms of extended stay on the ISS, we do observe significant atrophy of the muscles. And as a result, there is immense need to understand the mechanisms which are involved in the muscle atrophy and hopefully also develop interventions. And in this regard, model organisms are essentially important because it allows us to do quick experiments. And our focus has been to use this nematode as a model to look at any alterations in the muscle strength. So briefly, this worm is transparent, and the advantages of this worm in terms of the spaceflight experiment is that it lives only about 3 weeks, and you can generate from essentially eggs up to adults in about few days. So this amount of short time involved in producing all the larvae is significantly useful in looking at these changes in the physiology of the worm. And also we have significant amount of tools in terms of genetics and fluorescent reporters and so on, which really makes this organism a powerful model to look at how space flight affects the affects the host physiology. And if you look at the previous data from experiments, right, it shows that you do observe significant reduction in the expression of the muscle genes. And also studies have shown that the overall thrashing rate is also reduced. So all this leads to the hypothesis that in worms which are bred on ISS, we do expect some changes in the strength of these animals. So this has been the overall hypothesis of this Micro16 project. And in terms of the specific objectives, we have essentially engineered essentially engineered a new hardware which allows us to probe the strength of these worms. And the idea is to use this hardware to look at any alterations in the strength and correlate those alterations with any changes in the gene expression. And we would like to do this across different generations to see if the worms are undergoing any short-term and long-term adaptation on spaceflight. So the experimental design looks as follows. So as you can see on the top right are the bags in which the animals are being grown. And the idea is that we'll transfer aliquots out of these bags at weeks 2 and 4 and 6. And along with this transfer process, we'll also extract samples out of these bags and look at how the animals are behaving. And again, the idea here is to measure the strength of the worms, which I'll discuss on the next slide as to how we do that. And after doing this transfer and the recording of the images, then the idea is to bring the samples onto Earth to essentially do RNA-seq. And in terms of strains and the diet, we are using a strain, and we're also using this one, which is a model of the muscular dystrophy in which we expect the strength to be all decreased. And in terms of the media, we're using an axonic media to grow all these worms. And on the right, you can also see the actual hardware, which I'll describe on the next few slides. So this is the principle of the technique which we're referring to as NEMA-Flex. So the technique of NEMA-Flex allows us to measure the strength of the worms. And the basic idea is illustrated in this picture on the right, in which you see that the worm is deflecting all these pillars. So as the animal is crawling, it's encountering all these pillars and it's pushing them. And the experiment approach works in which you initially load these animals in this arena of all the pillars in this transparent device. And these pillars are special in the sense that they're essentially anchored on the roof, and there's a clearance. As a result, these pillars as a result, these pillars are allowed to deflect. And we watch the deflection on the microscope, which allows us to back out all the forces all these animals are exerting. So this is the basic principle of the NEMA-Flex approach. And at the time of designing the entire study, and we were thinking about all these questions, which is in terms of first, always in terms of spaceflight experiments, the initial step is to how to grow these animals properly. So as a result, and we were thinking about the diet, how much should be the starting animal density in these bags, and how much of this volume we need to transfer across weeks. And as I discussed in the earlier slide, these animals are laying eggs, and these larvae also hatch as well over time. So that means in this bag essentially have a mixed aged population. So it's important to understand because the strength depends on the age of the animal. It's important to understand the actual composition of these animals in these bags over time. And in terms of the NEMA-Flex as well, because we are interested in measuring the strength of a specific age. So the open questions are, how do we sort and identify animals? How do we reduce the hunt time, because it decreases the amount of time the crew has to spend to record all the pictures. And then we also have other questions which are more specific in terms of the hardware about how to load the animals and so on. So in this presentation, I'll not be able to address all of them. However, I'll be able to give you a quick overview of the approach which we use to address all these questions. In terms of the diet, I already discussed that we're essentially using the axenic diet, and the starting density was optimized to be 1,000 far away, and the transfer is being done every 2 weeks. And the data here on the left shows that after every 2 weeks of transfer, our numbers of adults are almost identical, implying that the whole protocol is optimized to ensure that the adults are being propagated healthily across time. And the data on the right is showing the thrashing frequency, which is an indicator of the swim health. And again, all these experiments were performed on Earth to essentially optimize the protocol of growth in these bags as well as the transfers. And here is the actual engineering aspect of the hardware. So as I mentioned earlier, our intent was to essentially sort or identify animals of the same age. And our approach was as shown in this design of the device on the left, in which we inject animals. And the idea is that these animals will flow along these channels and enter into these chambers. And this chamber have all these pillars, which allows the animals to deflect them and therefore allow us to record the strength. However, the important thing in terms of the design was to make sure that the animals were of the same age. And to achieve that, we introduced at the entrance of this chamber, this other channel, which allows only animals of a specific length and size to actually enter. So if I play the video, you'll see that the animals are being introduced into the channels, and then at some point you'll see an animal entering into the neck, and you'll also see that the larvae are also entering. And interestingly, we added additional channels downstream, which allow us to essentially use flow to remove all the progeny and the eggs. So this was the engineering approach which we took to ensure that as much as possible, we are able to record strength of the same age of the animals. And in terms of additional hardware, so here is essentially the HemofLex device, and we used essentially an approach which allowed us to trap these animals at a specific amount of the flow rate. And once the animals are trapped at this flow rate, we increase the flow rate to introduce these trapped animals into the chambers. And again here, and we had to do extensive optimization of the flow rates and the alloquads to ensure that we are obtaining enough number of individual animals in these chambers. Again, this entire project really depends on obtaining good pictures of the rims of all these pillars, because the forces are calculated from those deflections, right? So as a result, optimizing the hardware was also an extremely important step. And the other interesting aspect of this project is that and we were obtaining significant amount of data in terms of these pictures. So all the data was being stored in this external hard drives, which were again being brought to Earth to do the analysis. And a significant aspect of the project involved training of the crew to ensure that the astronauts are aware to adjust the illumination and the focus to obtain high quality pictures. So we essentially provided these reference images, which allowed the crew to be trained on how to obtain the proper in focus pictures of these animals which are actually crawling. Our software has difficulty analyzing deflections when these organisms are executing these kinds of very abnormal shapes or coiling postures, or sometimes if we have different animals being present, then the software also has difficulty. So as a result, both the engineering of the device and the optimization of the illumination and the focus, all of these were crucial aspects to ensure that we get high quality pictures. Here is the crew which did the experiments and a short snippet of the astronaut doing the transfer and the injection. Again, as I mentioned, this experiment was launched early, in February of this year. And right now we have the videos which we're analyzing as we speak. And as I mentioned, we're also looking at the gene expression of these samples. So essentially, and we have a team involving Hunter and Craig and Tate, and this team is looking at doing RNA-seq of these samples. And at this time, I would like my PhD student Hunter to elaborate on the how the RNA-seq is being how the RNA is being extracted and the procedures which we're using. Hunter, are you on the call?
Yes, I'm here. Hi, everyone. Glad to be here. I'm excited for this amazing project. So I guess, Dr. V, if you'll transition to the next slide. Okay, so we're interested in doing RNA extraction on these animals that we received back, or that we have received back actually at this point from the International Space Station. So to do that, we wanted to develop a protocol that would allow us to isolate the animals and then do this subsequent analysis. But the caveat to that is that these bags are filled with thousands of animals, and we don't want to use the entire bag to do a single sequencing analysis. That way, in case something goes wrong, we can go back to these bags and have those preserved samples. So we wanted to optimize this protocol for the minimum amount of worms that we could get out of the bags to do the RNA sequencing. So here's just in brief what we've developed so far. So once the bags are returned to us, we can then cut small sections of the bag, which allows us to thaw a tiny portion of the sample and preserve the remainder. And then from there, we assess how many adults we have actually collected from that bag. And we've done a series of analysis to see what the optimal number is, and we found about 300 to 400 adults is sufficient. So those animals are then transferred into a small aliquota buffer and then lysed through a series of freeze-thaw cycles using trizol reagent and liquid nitrogen. From there, the RNA or the lysed animals are then put into a spin column kit that we have sourced from Zymo Research. It's a direct thaw mini prep kit that allows us to elute the RNA from these animals. And from there, the extracted RNA is sent for sequencing, where we can look at the transcriptomic profiles of both the wild type and DIS1 animals on flight and ground. So this is just a summary of our optimization on pre-flight bags. We have not done this on our flight samples. And it shows in the final 4 columns that we are able to isolate using 300 to 400 animals, both a sufficient quantity and concentration of RNA for our RNA sequencing. And I think we're really excited to apply this protocol to our flight and ground samples to help us better understand the differences in muscle strength that we expect to see.
Thank you, Hunter. Yeah, so overall, I think this project had significant outcomes in terms of training of students and papers and collaborations as well. And this device, which is being used to record strength of the animals, has led to other spin-off applications in terms of looking at age-related declining strength, as well as muscular dystrophy. And I'm also excited that we are still going to be involving these kinds of devices because of our next project, which is looking at how the microbiome is going to affect the host physiology. Again, this is truly interdisciplinary team involving different involving different PIs, and our goal is to look at not only changes in the expression level of genes, however also to look at all the different tissues, and finally also look at the entire host in terms of the physical strength and the locomotor activity as well. So with that, I would like to thank the opportunity, and I'm happy to take any questions. Thank you.
I have one question here, so maybe we can answer that before we go into the breakout rooms. Have you considered using anabolics in space in a future study in a mammalian animal model?
I'm sorry, could you repeat that again?
Have you considered using anabolics in space in a future study in a mammalian animal model?
I'm sorry, I'm not aware of this word anabolics. Would you expand on that?
If anybody, whoever wrote this question, if you can expand on that, please do so. Hormones.
Okay. So the question is, how can we look at hormones using a mammalian model system in spaceflight?
Yeah. Yes.
Yeah, I don't think I'll be able to answer that because I've not had enough time to think about that. Again, if you're thinking about looking at individual cells or actually organisms, it could really change the actual design of the hardware and so on. So I'm sorry, I'm not able to answer that question.
That's all right. Thank you so much. We are a little bit past time here. I'd like to thank all of our speakers. They're all great talks, and I want to thank you all for your time. And right now, the speaker presentation portion of the workshop is over, and we want to open